मेरा स्पीकर बंद है।
Adesh, you can start.
Yes, please.
Yes. So good evening, everyone. We are delighted to have you join us for today's enlightening webinar, which is Sperm Walk, the Marvel of Motility. We all know that sperm is the half of the gamete in the embryo, and it plays a very crucial role in its embryo development and getting successful pregnancy. In this microscopic world of reproduction, there are few phenomena which are captivating and crucial as the journey of the sperm. This session will take us deep into the science of sperm motility, and to speak on this topic, we have esteemed faculty members, and we have renowned speakers with us to help us guide and help us understand how the sperm motility works and what are its function. Before we start, I would like to welcome and introduce our founders, Dr. Ved Prakash sir, Dr. Sanjay Shukla sir, and Dr. Charudha Joshi sir. I'm sure they need no introduction in our field, and everybody on this platform already knows them very well. They have been very kind to us, younger generation of embryologists, by sharing their knowledge and experience. Um, I would request to Dr. Charudat sir to say a few words before we move on to next session, please.
Thank you, Desh, and very good evening to all. Yet again, it is my privilege to offer you to introduce you to another upcoming webinar on male factor from iHera. This is an ongoing online learning platform, as rightly said, for our youngsters, for our young generation of embryologists to share their knowledge, to gain the knowledge, and to understand it better. And now we all know male factor has been considered and it is given its due importance, which was neglected earlier. And lots of activities, lots of research, lots of work is going on. And we are getting the regular updates also. So we definitely need such kind of interactive platform where we can understand, we can learn what happening in the field of andrology. So, without taking much of your time, it's once again, it is my honor to welcome all my esteemed faculties, welcome all of you, all delegates, and we can just be ready for listening about the male factor from the stalwarts. Thank you.
Thank you so much, Charudat sir. Now I would like to welcome and introduce to our conveners, Dr. Devin Patel, Dr. Hetal Shukla, and Dr. Durai. A warm welcome from all of us here. And Hetal sir, I would request you to speak a couple of words for us, please.
Good evening, everyone, and uh, everyone is now know about iHera, and iHera has already brought an andronet into the action, and andronet, since andronet has come into the action, male factor is given more emphasis academically, and it is a great pleasure to inform you about Andronet becoming more and more active, coming with more and more kind of academics into the action. So, uh, yes, today's uh, discussions will be definitely targeted on the uh, motility of the spermatozoa, whether whatever the purpose of the sperm, purpose of the sperm is only one, that is to fertilize the ova. But there are different ways: in vivo fertilization or uh, in vitro fertilization, maybe. But sperm's travel distance might be reduced, right? There are three instances. One where naturally, I mean, from reaching to the fallopian tube, or in the fert dish, in the fert medium, their sperm either has to reach to the oocyte with a very little distance, but yes, it is a critical one. And when embryologist is selecting the spermatozoa, uh, probably motility is one of the important and the most uh, 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 preferred uh, tool to select the spermatozoa. So motility, of course, matters a lot. What is the science behind? Let us try to understand what could be the challenges, how the challenges could be mitigated. Probably today's journey of this discussion will help everyone to just understand that. Thank you, Ihera, and thank you, everyone, being part of this. Thank you.
Thank you so much, Hetal sir. Now, let us begin with our program. I would like to introduce Dr. Akash, who will be speaking on the topic guiding the sperm walk, chemotaxis, and rheotaxis in fertilization. Dr. Akash has over 15 years of experience in embryology and is working as head of QA/QC in embryology at Nova IVF. He's a ISHRE certified embryologist and has been topper at it. Also working as assistant professor, visiting faculty at Aspire. He is a managing committee member of ACE from 2022 onwards, and has been a member and on a scientific committee member of IHERA, a special interest group of IFS, and all other embryology bodies in India. I'm very sure it would be very interesting to hear from him on this topic. Over to you, Dr. Akash, whenever you're ready, please.
Before starting that lecture, I would like to say something about that. We forgot to introduce our co-convener also, Dr. Jay Sinde. He's a very good embryologist. You know that more than, I think, 10 years experience he has. And second is Dr. Siddhar Morvadia, and he's also a senior embryologist in Hyderabad. And third is Priyalsa. So all of three are taking care of this webinar, and they are joined us. I think since our Android started, they are doing very good job, and I think they will take it forward that Android what we mean. Thank you so much. Thank you. What do you? Yes.
Thank you. Thank you. It's a pleasure to be a part working alongside you guys. Thank you so much, sir, from all of us. Dr. Akash, we can start whenever you are ready, sir.
A very good evening to everyone. Thank you to Andronet, a subset of IHRA, for what on the topic I would be speaking, which would be guiding the sperm walk, sperm chemotaxis, and rheotaxis. Just give me 1 minute, I'll start my screen sharing. I hope the screen is visible.
Yes, sir, it is visible.
Thank you. Thank you. So we are speaking about the sperm chemotaxis and rheotaxis. We all know that the sperm, human sperm, takes roughly around, say, between 2.5 and 3 months somewhere for the maturation to complete over there. And post that, what we do see is they undergo a maturation in the epidermis. So what do they actually acquire during the epidermis maturation? First is the important property of motility which they acquire. This is very, very essential for the journey. I mean, a sperm which is deposited in the lower female genital tract without a motility, obviously there's no chance of it fertilizing the oocyte over there. So the sperms which are deposited in the lower female genital tract as such have this big task of reaching the oocyte. They have to travel up the female genital tract, And for this to happen, post liquefaction, they have to undergo a process called as capacitation, which you can see over here. So what does capacitation do? So capacitation is a process whereby a loss of cholesterol esters happen in the cell membrane of the sperm cells as such, and thereby there increases the fluidity in the sperm membrane. So this can now respond to even additional changes as well. So this is what the capacitation helps in. So it results in the membrane becoming more fluid, and it even gives the sperm the ability to fertilize. And then our sperms, which are there trying to reach the oocyte as such. So capacitation usually happens in the lower female genital tract, removal of the cholesterol membranes, as such. And this results in something what we call as hyperactivation of the sperm. The sperms remain in a quiescent way inside the male genital tract, and even when they are undergoing maturation, there are changes in the pH, the bicarbonate buffer as such, which is present in the female genital tract, plus the osmolality changes, which happens from the epididymis, caput epididymis, to the vaginal tract, which helps in the hyperactivation of the sperm. But what use is the motility if it is not guided? It is like you have lots of energy present over there, but that is not being channeled. So this channeling of energy is very much required. So this happens even through a process of capacitation. The ability is gained over here. So here, if you go look at the molecular basis of it, capacitation, again, you'll see your cyclical nucleotides as such. You see AMP, which is increased, your cyclical AMP, which results in the capacitation process as such. I'll not go into much details of this. So what are the major challenges faced by a sperm? So you see the hostile environment, the pH of the acidic pH of the vagina, which definitely would result in killing of the sperms if they stay for a long time. A very, very tough competition from millions of its counterparts, which are present in hundreds of millions in a normal individual, normosospermic individual as such. And then you may have motility to go ahead, but you don't know which direction to go. The one what I was saying. So is it just the brute numbers, just the sperm being in millions and millions of them, and randomly one of the sperm happens to stumble upon the oviduct and then goes into and fertilizes the oocyte as such? Is it that? Or is there something, a stronger force which is guiding it as such? So the progressively facing motile sperms, they face a challenge. What should we do? Where should we go? That's the question that they ask. It would be very easy if we can compare a sperm to a vehicle which has got movement but no direction, so guiding force as such. So what gives the cells this direction? So we find out that it's not just it happens at random chance, but there's few guiding angels, if we may call them, signals that guide the sperm in their journey towards their destination, which is the oocyte. And these signals, we'll see what are these signals in the future of this webinar as such, and how these are controlled. And what's really fascinating is this at the end of the day seems to be controlled by the female counterpart itself, these signals. So after all, it turns out males don't have any choice if we equate the males to the sperms. So once again, so the challenge which now remains is the third one, which we are dealing with, which is how does the sperm guides itself? So it guides itself by three various mechanisms. The mammalian sperms guides itself by three various mechanisms to reach the egg. The first one is the chemotaxis, where it's the ability of the sperm to be responding to the chemical attractants, which is progesterone primarily from the cumulus cells. Thermotaxis, where they respond to the temperature fluctuations. We all know that the temperature of the female in general and the genital tract as such in particular increases around the ovulation time. We see that. So that's a guide for this. And then the rheotaxis, which is the ability to respond to the fluid flow or the shearing stress. So these signals will help the sperm and guide them in achieving the path which will show it nearest oocyte. And thus successful fertilization will happen. So if we put it in a pictorial way, we can see the sperms which are mature in the epididymis released by ejaculation, and then they undergo hyperactivation by capacitation, and lastly the chemotaxis part. So one of the things which we did, capacitation happens spontaneously in the, say, it happens with the aid of the lower female genital tract, how would it happen in the ART setup in the embryology lab? So what we do a procedure commonly called as swimmer that usually helps in capacitating the sperms. So we'll be seeing each of the taxes in specific. The first one, what we are going to look at is the chemotaxis. So chemotaxis, the name itself shows that it is the response of the sperm in turn to a chemical signals. So whenever there are taxes happening, three things should happen. First, there should be detection of the stimulus, which is the signal. In this case, the chemical signals or molecules over there. Next, a signal transduction machinery should be there. Such once it is detected, it should be able to transduce the signal. And in this case, post that deduction, the change which we are expecting is a change in the motility direction, not change in motility per se, but change in the motility direction. And lastly, there should be an adaptation to the signal. Otherwise, once activated, it should not be continuously happening. Otherwise, it can result, say, the sperm is moving towards one direction, and then the signal says to move left. The sperm may turn left, but the signal should attenuate, otherwise it will continue to move left. So that is also very important. So these are the three basic parts of any taxis to happen. So motility was observed way back, some historical facts over here by Anton von Leeuwenhoek. He could see that this bacteria which are present in collected from a oral sample of a person with dental plaques. He could see cocci, fusiform, and spirochetes which were moving. And we know that the spirochetes have this typical corkscrew type movement as such. And discovery of chemotaxis also new, not new, sorry. It was discovered way back in 1883, one of the papers presented by Engelmann et al., where he showed that the bacterium movement is not random, but rather it is guided by chemical signals. And as such, in gametes, it was shown by Pfeffer in 1884. It was basically, he has shown in that the fern gametes, where external fertilization takes place, that these signals, as such, help the sperm in guiding their way towards the oocytes. So coming back to a main part of the chemotaxis, so this involves your chemoattractants. So the first part where I had said that there should be a signal and that should be transduced. So what are these signals that we see in human sperms as such which they respond to? So for chemotaxis to happen, the first and foremost would be the progesterone. These are usually secreted by the cumulus cells as such. The progesterone secreted from the cumulus cells is in the range of picoliters, so picomolars. So it is that low, 10 to the power minus 12, something of that range.
I think he lost his connection.
I also think the same.
Yes.
Mm.
We are sorry, extremely sorry for this. I think Dr. Akash has dropped out. We'll try to get it sorted as soon as possible.
Nidhi, can you please call him? Just try to find out what happened.
Yes, sir. Yes, sir. So till that, we can have a discussion regarding what we have just now. Practically, how the capacitation and we are doing it at other
Yeah.
centers, sir.
I think Hetal
Hetal, sir, if you could enlighten us about it. Hetal, sir, are you there?
Hetal, Dr. Hetal. Where is he?
Deven sir, would you like to discuss anything or start a discussion?
Yeah, rather, I was about to pose a question for Hetal Shukla, but I think he is not there. So maybe I think Ved sir also can answer this. So
Yes.
basically, Akash was explaining, there are different type of taxis. So one of them he was talking about chemotaxis. So like microfluidic devices are there. They are also working based on some progesterone, then some temperature and all the different taxis. So is there any device, if we know, which is having all these together? So basically in uterus, maybe these all signals are working together. When we do microfluidics, there is only one taxis we work. So if you know, is there any other device which is having all these three together?
I think there is one on LinkedIn. I have seen that one person is talking about Riotech or something like that device is available. And it is working on all the three, the Rio, Chemo, Thermo, all the three. Uh, I have no actually exact idea, but definitely when he explained all about these things, that particular device is working on that all the three parameters. Riotech, I think Riotech or Riotex. It is
Okay.
name, device name.
Yeah, I
And I
also
from Egypt guy or Egypt, I think he is from Egypt. Egypt.
okay. So I think that is the future, sir. Like, because see, we are working with one different techniques. Like if we do density gradient or if we do stream up, then microfluidic. So these are like max. So then these are
Sir,
working.
sir, sorry, I am disturbing, sir. Please allow Akash sir to enter the.
Yeah, please.
So they're not. The host has to allow him, sir.
Yeah,
He's trying to log in.
tag team, please allow him.
Hello. Can
Yeah.
you hear?
Yeah, yeah.
Yes.
Sorry,
You are back.
I lost the connection. It was still which slide? I'll just share once. Let me share. Till which slide was I audible?
Up to 10th, 11th slide.
This was it this
Yeah,
Yes,
I
sir.
think
We were on
yes, sir.
chemotaxis discovery. Yes, sir.
progesterone?
Correct.
Okay, fine.
Yes, yes.
So sorry for the distance and coming back. So I was saying about progesterones, which are usually secreted by the OCCs as such, and then a hydrophobic non-peptide molecule, which is secreted by the mature oocyte itself, and there are additional molecules which are proposed to be as such to be your chemoattractants, which are CCL20, your atrial natriuretic peptide, and natriuretic. The point to be noted over here is these usually molecules and progesterone and the a range of certain millimeters as such. Is this feature that we see in chemotaxis, is this specific to humans? No, we see that this, as said, this would be their chemotaxis feature way back where external fertilization would take place. So thereby it's very essential, guides them, and it is species specific. But what we see as evolution has happened, there has been internal fertilization taking place. So what is seen is the key is specific between the sperm and species specificity. So the follicular fluid and other fluids which are carrying these chemoattractants as such, and the progesterone works as such a chemoattractant for many of the species. And next thing would be that the progesterone acts. In what way does it act? Is it the gradient of the progesterone which causes it over there in time or in space? So what is usually found is that it is the temporal changes in the gradient concentration over time which is important rather than as this. So this has importance in terms of how it is going to locate the oocyte. So what is the chemotaxis response to the gradient as such? The behavioral changes usually we see in the sperm motility in response to these gradients, they usually involve a change in the motility pattern. So what they say is there is a change in terms of the frequency of the turns and the hyperactivation events which are going to result from them. So a sperm which is hyperactivated and suppose there is no gradient as such, it would be moving in random. Once there
Sir,
is a
there is still some connectivity issue. I think so, sir. If you could look into it, sir. It's getting
okay,
Your voice is breaking in between, Doctor.
yeah, voice is breaking. So just see.
just give me 1 minute. I think this. Just give me 1 minute. Let's reset the. It's audible, right?
Yes, it is audible.
Hello.
Doctor.
Yeah, sorry. So we see the chemotaxis respond to the gradient. So this behavioral changes what we see in terms of the hypermotility which the sperm acquires, and more importantly, it is so the turns get reduced. So what happens is a sperm which is going to be there, which is going to move around in circles as such, you can say, once it sees a gradient coming in, so it will orient itself around there because from the is present temporally, the turns reduce. So the turns reduce. So thereby, as such, as long as the gradient is present temporarily, the sperm continues to swim along this gradient. And this applies usually for your chemotaxis as temperature gradient, your thermotaxis as well. And this gradient is something which is present in the E. coli bacterium. This is not something which is unique to the mammals or humans as such. So this is something which is present since long. Only thing is it is seen in the sperm cells, which is job to be performed. At the molecular biologists as such, we should say we'll see things in microscopic at but sperm cells. What actually causes this chemotaxis at a molecular level, that is not very clear. But there is activation of a transmembrane ligand, which usually results in binding of with this receptor as such, and this cellular asymmetry, which is the turn happens. So the sperm continues to swim towards the gradient. Our best understanding of this comes from a sea urchins in the past. So they have a CHI-A, CHI-A, and CHI-Y. These are the various molecules which are present in the bacteria. Your basic as such for sperm chemotaxis, which is mediated by progesterone in humans, this was elucidated by Maria Tevis and the team over here. And they have shown this the schematic representation as such. You can see the progesterone will bind to a transmembrane receptor, the progesterone receptor, which is present over there, and it will cause an activation of the transmembrane adenyl cyclase activity. So basically, it will result in an increase in the cyclic AMP levels. This will activate your protein kinase A level, which will cause protein transphosphorylation as such, and will cause an initial release of the calcium stores. I would say initial release because it's the part one of this. This will activate the calcium channels which are present and cause further release of, the further increase of intracellular calcium levels. The first rise, which was present with the intracellular calcium stores the release, and the second one is from the external, the calcium sweeps into the cell as such. Their cyclical GMP again rises. So it was CAMP first, now which rises, and then protein kinase G is activated as such. And still now it is not properly completely elucidated how this results in the turns. What are the changes which happens in the axoneme? They are not fully elucidated as far as I know about this. So this is the missing link. But then they have shown that the calcium increase which happens, this is linked to the axonemal changes. in terms of representation, where we see the phosphorus interaction with the receptor, your TMEC getting activated, your PK getting activated, phosphorylation release ending up in the release of the calcium intracellularly. Next, again, there'll be activation of the cGMP and finally the second calcium influx. And this will result in the behavioral changes by binding of unknown ways to the axoneme and a change in the sperm direction. And they have in this paper presented that when your progesterone is present at a lower level, where it's absent, your CAMP levels are of this range in the one where they are more than double once progesterone is present. Similarly, for the cGMP, they have shown that it is present in lesser volumes as such in the cell, but then their concentration increases much more when they have been exposed to progesterone externally. And this is in terms of how over time, if they are exposed to calcium, the calcium levels which are present intracellularly are at a lower level, but once they are exposed to somewhere in the 10 picomolar range of progesterone, the calcium inside the cell increases. So all these indicate how they have deduced that it is your progesterone, which as a transmembrane ligand will increase your calcium levels as the final step. So this was what I was saying, that initially it will be going round and round, but once it has some guiding force, which in this case I was speaking is progesterone, will guide it towards the egg. The sperm is guided towards the egg. The turns are suppressed actively. The axonal structure always the dyneins which are going to the motors, which are powering the movement of this. This is the part which is missing, how the calcium increase gets coupled to this dynein selectively at the place. So if we put chemotaxis in short, it is a short-term force in terms of acts over a few millimeters. Progesterone is the well-connected one in terms of well-illustrated mechanism, how it happens, and the lesser number of turns, and thus the sperm moves straightly up the gradient. The next part is the rheotaxis. The sperm or any organism where they'll be moving in response to the flow which is present in the liquid. So again, it is seen in many bacteria as well. It is seen in aquatic animals, including the fishes, where it is called as positive rheotaxis, where they hold themselves against the flow. This is a common thing what we might have seen whenever we put a drop of semen and watch it under the microscope, put it a cover microscope immediately, we see that the fluid may be moving in the one direction, and majority of the sperms will try to orient themselves against the direction of the flow of the fluid. And this can be much more easily visualized whenever we see the washed sample as such. So what is happening over here, when you put the raw semen sample, they are not that much capacitated. But once you are seeing a washed semen sample, this property of rheotaxis we can visualize directly under the microscope. And there are some recent papers which say that it is not just limited to living beings, but it can be seen even in something called as self-propelled particles, which are gold platinum nanorods. So what this tells us about rheotaxis is it's more of a physical phenomena, a biophysical phenomena, if we may say, in terms of sperms, rather than something which is connected actively. And it is a passive process. And these were demonstrated first way back in 1961 by Rothschild and others, where they showed that the human spermatozoa can hold against the flow. And the importance of rheotaxis is that it acts in a long scale, like it acts over centimeters. So in mice, it has been very clearly shown that there's the oviductal fluid which is secreted within a few, and it will tend to couple this with the uterine peristalsis. It will have a downward flow as such down the track. Thereby, the sperm will be positioning themselves against this flow, something what we call as positive rheotaxis, against the flow, and they'll be swimming against it. So this is a cue which the sperm search over a long distance, over a distance of centimeters as such. So the point which I was telling in terms of this being a long-term distance as such, acting over a long distance, it's a biophysical phenomena and it is passive. The important part over here is it does not consume energy as such, whereas your chemotaxis and thermotaxis consumes energy. Rheotaxis as such is a long-term guiding force and it does not consume any energy per se. So the positive rheotaxis, which I said, is the ability to orient against the flow. And something interesting over here was there was a study where Demartin and Zafrani, they have shown that as such, the sperms they have isolated based on the property of rheotaxis. And what they found that the sperms which are able to demonstrate this property of rheotaxis, they have a better morphology and the chromatin status is also better. So thereby, nature is probably selecting sperms by these rheotaxis which have better morphology and which are having a better chromatin status. So the chances of fertilization and the chances of successful progeny being formed and they having better health is over there. So whenever we are trying to maybe replicate this rheotaxis in future in the lab, then that not only just gives us a better sperm in terms of fertilization status, but then it may even give us a better result in terms of a progeny health, which is a very important point to consider. So other than this, there's something called as thigmotaxis. Your the thigmotaxis is something which is a bit related to rheotaxis. That the sperms tend to search corners. Good sperms tend to search corners, and if as such, they are much more motile and rapidly motile when they are going along the corners. So this is already demonstrated as such. See in our lab is whenever we load a semen sample, process semen sample as such, the sperms which are most actively motile, if you are making an elongated PVP draw, flattened one, the sperms which are more the corner and they stick to the corner and there they'll be progressing much more rapidly. So this is called as thigmotaxis over there. And what is the correlation in vivo? The sperms which are more motile and more better, they will be moving along the grooves, what we call as crypts present in the female genital tract. So again, thereby by creating these grooves, and if you are going to select the sperms which are coming towards the thereby maybe we are selecting a sperms which are definitely better in terms of fertilization potential and in terms of the chromatin status as such. So this is a representation of in terms of what the sperm rheotaxis is. It's a major determination over long-term distances. It is a biophysical process. It's a passive one, and it works over long distances, and thus is an important guide when it comes to guiding the sperm. Pictorial depiction of how a rheotaxis, thermotaxis will be looking at factors which guide the sperms which are deposited in millions over here. Then some of them survive and then they'll be able to pass up the cervix and then they get capacitated. Then they move along still along the genital tract in terms of your pattern, which is aggressively motile forward moving. And then even against the sperm, against uterine peristalsis. So nothing but rheotaxis. As such, with uterine peristalsis, they'll be coming near the oviduct, the opening, and then near there, probably that's where your OCCs, if it comes over, and it will be secreting the progesterone, and there it begins to have the action as such. Even then, it would act in the vicinity, and once it reaches over there, the progesterone and the other chemoattractants, what we had said, will start to have an effect over there and guides it towards the mature oocyte which is present. So this is the slide as such, which shows a combined effect of your chemothermo and rheotaxis. But there are certain additional points as well, which I would like to highlight. What happens over there, which increases the chances of fertilization naturally, if I may say. Capacitation usually happens, just roughly 10 to 20% of the human sperms. So that's quite less. You're reducing the number by 10 times over there. And whenever mating happens and the sperms get deposited, it's not compulsory that the mature oocyte with the OCCs is present in the oviduct. So how does nature increases this? So what it does is not all the sperms get activated at the same time. They get activated over a period of time. So thus you'll have a pool of sperms which are getting continuously activated and which are migrating up the female genital tract. Because once they get activated or capacitated, what we say, the active lifespan is just order of few hours. So the oocyte may not be there. So to increase the chances, the probabilities of the sperm to be present when the oocyte is over there in the oviduct. The nature does this trick whereby the capacitation happens in a series, not at one time, but in a series of waves as such, and thereby the sperm chance of finding a mature oocyte in the oviduct. So this was the, and next, this was the paper which showed recently, we talk about symbiosis and everything. We see sperm as competitors to one another when they are present in the lower female genital tract. But this paper showed that the sperms may not be always competitors to as such. What they have shown is when the sperms come from the same male source, they tend to group together whenever they are moving from the low viscosity to high viscosity media, something from where they are traveling up the female genital tract. Then in these cases, few of the sperms group together and move ahead, and thereby the motility as such increases by 50%. So how much of this effect has in vivo, that is still yet to be deduced completely, but definitely it can be seen that the sperms are working symbiotically among themselves. Thus, more motility, more the chances of going to the oviduct, more the chances of finding the mature oocyte over there. Another interesting fact is this paper shown that this symbiotic kind of behavior happens between sperms which are from the same male partner as such, but not from the different male partners. So supposedly from different male sources, then this symbiotic behavior was no more shown to be present over there. Your sperm thermotaxis additional to chemo and rheotaxis, and the gradient which can be as low as 0.014 degrees centigrade per mm. This is per mm, mind you. So it's so small a difference also, the sperms can sense it over there. So additionally, there were certain other points which were there, like what is being now told as there was a paper which shown there's a cryptic female choice, that the oocyte exert effects in terms of even which sperm which reaches the OCCs and the oocyte membrane zona pellucida as such, which of them will be able to fertilize. Still unknown mechanisms, how it happens. They have clearly found that there's a difference over in terms of which sperms are able to reach the oocyte and which are able to fertilize it. Mechanisms still have to be elucidated. Fine, well, we have seen what happens, how a sperm is guided in nature in terms of the how it is able to reach the oocyte, but what are the potential applications for us in infertility? Well, one is we can better diagnose for male fertility. The sperms which are able to have this post-capacitation better reactions, they can be better able to have fertilization potential, and we can counsel the patients accordingly. Micro based on chemotaxis and rheotaxis and even thermotaxis can be designed as such, where they can be shown that the sperms which can be separated based on these properties of them, they have one well which has progesterone and the other does not, and the gradient will cause them to separate over there. And again, a flow may be present in one of the wells, one of the sides, microgrooves, and it may not be present in the other side. So these are all ways by which you can have a separation of sperms, and thereby you can select also sperms which are better in quality, the way nature probably selects the sperms for better quality, and that's how we can improve our sperm selection as such. And when we apply this on our ART procedures, microfluidic devices will be available very soon, which will definitely have a role to play in this selection as such. And thereby you are increasing the chance of not only just fertilization, be it ICSI, you are even increasing the safety profile of the progeny as such. And thereby you're reducing the gap between the in vivo and the in vitro. So thereby harnessing the power of your chemotaxis, rheotaxis, thermotaxis, and others which may be elucidated in the future, you can close the gap between what causes in vivo and in vitro differences as such. So what is your take-home message finally? The journey of the mature sperm in the female genital tract begins with capacitation. Capacitation results in a hyperactivity sperm. Not only that, it even confers the sperm the ability for chemotaxis, thermotaxis, and rheotaxis. Chemotaxis is an active process which is mainly mediated by progesterone from tubular cells and also from the mature oocyte itself. Rheotaxis is a passive phenomenon and which is present over long distances, over centimeters as such. There are even other sperm behaviors which aided it in the successful fertilization as such. There are potential applications in terms of better diagnostics tools, where and even better sperm separation and selection tools, which have the potential of increasing our chances and even the safety profile for the progeny. Thank you for patient listening, and once again, sorry for the disconnections in terms of the network. Thank you.
Thank you, Dr. Akash. It was very in-depth and very good presentation, good topic. Explained very beautifully and nicely. Ved sir, I would like to take your opinion here. If we can have maybe a couple of minutes for a couple of questions in between, or we start with the second session because we have lost some time in between.
Akash, will you stay for some time because Dr. Murik is already waiting for his talk.
Fine, sir. I'll be available. No issues.
Yeah.
Okay. So everyone, we can maybe hold on to your questions, and we can connect after the session with Dr. Akash to get your query sorted. Meantime, I would request Siddharth to take over and start with the second topic. Siddharth?
Am I audible?
Yes, yes, you are.
So, introducing our second speaker, Merrick Gala, PhD. He's basically from UK. He has done PhD in applied mathematics from University of Birmingham. Mainly, Merrick.
Siddharth, can
Yes.
you start that CV of Dr. Murik?
Yeah, it's not visible.
It's not.
No, it is
Okay,
not
okay,
visible.
sorry. Okay.
Yeah, now it's
I hope
visible.
it's visible now.
Yes, it's
Yeah.
visible.
So sorry for that. So Merrick Gala, PhD. He has done his PhD in applied mathematics from University of Birmingham. So in bio, he integrates mathematical modeling and software with experimental methods to create new diagnostic and treatments for infertility, as well to as tackle wider healthcare applications using his comprehensive interdisciplinary approach. So as reading it, it seems quite interesting. So I hope his session will be way more interesting than this. Over to you.
Thank you very much. I'm just trying to share. It doesn't seem to let me while you're still sharing.
Yeah, I stopped it. Now you can.
Thank you. Okay, can you... Oh, I'm getting it. Can you see that?
Yes.
Yeah, visible.
Brilliant. Thank you very much. And thank you very much for the invitation to talk. It's great to be here and to chat to you. So, yes, as you said in your lovely introduction, I'm a mathematician by background, although nowadays I work in a very interdisciplinary way. So I work with an experimental lab. I'm also have an office based out of the Women's Hospital in Birmingham. So I work very closely with the team in the fertility centre there as well. So we kind of capture all aspects of what we can think about with regards to men in fertility. I'm going to talk to you today a little bit about sperm generally, but particularly about some of the work that we're doing around different ways to assess sperm and what we should be thinking about and how we can use these ideas that we develop in Birmingham to better understand sperm and use that in a clinical context. I don't have any conflicts of interest that directly relate to this work. We've got quite a lot of research funding in our group at the moment, particularly from what is now the Gates Foundation, but also some UK funders as well, but none of that is commercial or impacts what I'm going to say. So I'm sure you all know this, but it's worth always kind of reiterating it, that sperm are incredibly complex and incredibly cool cells. They're the smallest human cell. They have to propel themselves, but not only that, they have to propel themselves through a whole load of different complex environments themselves, you know, from the gel like secretions of semen. As that breaks down, they still need to be able to swim and then propel themselves out of that into cervical mucus, which is phenomenally complex as a viscoelastic fluid itself, and then all the way through the female and then penetrate the egg coat. So it's a really interesting, mechanically complicated task. We've got these kind of main pieces, as you will know. If I put my pointer, we've got the head of the sperm, we've got the mid piece, the principal piece, and this little tiny end piece, which, as I'll talk about a little bit more towards the end, is actually super interesting. It's not just a tiny little bit of plasma at the end of the tail. It's actually quite a useful and important piece that goes on. If we think about the numbers of sperm and actually what we're trying to what we're expecting to be able to do, actually we see why in itself why we've struggled so much in developing better ways to assess and diagnose male factors in fertility. You know, we're starting with about 100 million sperm deposited into the vagina. That's huge numbers of tiny cells, but only about 1% of those at absolute best are able to penetrate from semen into cervical mucus and then make it the 4 centimeters or so through the cervix into the uterus. So that's a giant selection mechanism in and of itself. And then by the time we get to the egg, we're only down at 10, maybe 100 sperm. So just thinking about the numbers, it's a really statistically complex problem. If we're looking at a sperm sample diagnostically, we're never looking at sperm that have made it to the egg because that sample has not been part of intercourse. We're always trying to measure populations of cells that aren't the cells we're interested in. We're never, or unless we're looking particular at ICSI, we're never able to actually look at the individual sperm that is the fertilizing sperm to be able to know what that sperm looks like. So really, we're spending a lot of time looking at a lot of sperm that aren't the ones that we're interested in. We spend a lot of time thinking about yield and number of sperm as being a key marker. But actually, when you think about it, that's very rarely the right way to think about sperm, particularly in the human. It's not about numbers. It's about how do we identify and understand those small population of sperm that are the really key ones out of all of those that could have potentially made it. I'd just like to show you a quick bit of data that kind of, in my mind, really motivates why we spend all our time looking at sperm and why we think more people should in the context of fertility treatment. So the first is a study from almost 6 years ago now from Fabrizio Horta out of the Temple Smith lab in Monash in Australia. So they took 2,500 IVF ICSI cycles from couples with unexplained infertility and then looked at their predicted live birth rates against female age. And as you expect, the older the woman is, the lower the predicted probability for live birth. What they also stratified against was male age. So these colours in each section here are stratified by male age for a given female age. And you can see there's significant decreases in outcome by male age as well, independent of the female age decrease. And pretty much as significant. For an older woman, we're going from, predicted probability of live birth from about 15% down to about 7% in the older male age group. So that's a really, really big impact on male age on outcomes. In terms of the individual sperm, this is a study that we ran partly from Birmingham in the UK with that called the HabSelect trial. So this was a trial comparing ICSI, standard ICSI practice to PICSI. So for those of you who don't know, PICSI is exactly the same as ICSI, except you select a sperm that binds to hyaluronic acid. So mature sperm have an HA receptor on the head of them. You swim them over a little dish that contains a coating of HA. The ones that bind, you select one of those and inject one of those into the egg. This was almost 3,000 couples across 16 centres in the UK. And the mechanistic analysis showed this essentially. So if you look at the red curve first, this is predicted live birth given the fact that there's a clinical pregnancy. So once you've seen a fetal heart, what is the chance that you will go to term plotted against female age? And in ICSI, for a younger woman, you see once you've got the fetal heart, you're very, very likely to go to term. And if you take someone that's around 40, you're only about 40% chance to go to term. ICSI is completely flattening that curve. So again, the only thing that was changed in these two arms was changing the precise sperm that was selected and injected for treatment. So to put those in numbers, if you took a woman that was 37 years old with ICSI and she got a fetal heart on the scan after treatment, she would then have a 40% chance of miscarriage going forward. Just by changing the sperm, you're taking that down to a 15% chance of miscarriage. So that's a really, really significant change just by thinking about which sperm is going in. So what's going on there? How can we try and assess sperm in a way that kind of gives us insight into some of these factors in a way that's better than we currently do? Well, to do that, we really need to understand what makes a good sperm and what the tasks are that a sperm needs to do. So the obvious one is that they need to be correctly assembled. So that means they have to have the right shape. They need to have a head, a tail, a mid piece, an end piece, all of those things that I showed you at the beginning. The DNA needs to be correctly packed. There needs to be the right amount of DNA. It needs to be not damaged. And then the sperm needs to be able to have all correct function. So it needs to be able to keep itself in homeostasis so that the DNA can't be damaged in transit. It needs to have the correct signaling. It needs to be able to respond to, as we've heard in the previous talks, stimulants such as progesterone. It needs to have its correct calcium signaling. It needs to have its correct metabolites. It needs to have the centrioles correctly present. Obviously, the centrioles are forming the spindle as part of the first division in the egg. The problem we have is that most tests to assess this are invasive. They involve blowing up a sperm head to look at the DNA, or they involve killing the sperm, fixing it and staining it to assess its morphology. And again, just as I highlighted at the beginning, they're only measuring populations of sperm. We're not measuring the individual fertilizing one. And that's complex. If we're thinking about sperm selection, then we need non-invasive insight. But really, if we're thinking about any form of sperm assessment, we need to be able to make these assessments in a way that is understanding all aspects of these components on an individual sperm, which includes things like motility, includes things like whether it's alive or not, all these things that you can't currently co-test for on single sperm. There are a whole load of tools out there. The previous talk touched on a bit of microfluidics. There's lots of other challenges. There are things, hardware approaches like looking at DNA fragmentation or reactive oxygen, thinking about microfluidics, thinking about IMSI or MAX. If you're wanting to do something where you're selecting sperm, you can put sperm against a viscous challenge. There are also lots of software approaches. People are doing a lot of AI on sperm to try and understand what's going on. So the question is then, why, given all these tools and all this information that I've just told you, are we not always selecting sperm perfectly? So what are we missing? What isn't part of the conversation at the moment that really should be part of the conversation? Well, really, we need ways that we can assess sperm better. So all these methods that I've talked about, most of them on their own have very, very weak prognostic value. A lot of people say we should always be doing sperm DNA damage tests, but actually most of the data shows that most DNA damage tests don't correlate with each other, let alone correlate with treatment outcomes, for example. Ditto with microfluidics. People can spend a lot of time designing microfluidics that cost a lot of money and do a lot of very fancy things to the sperm, but actually, if it's not changing outcomes, and if it's not changing meaningful outcomes, then there's no point in doing it. But we do know that changing sperm changes outcomes, and we do know from the data sets that I've just shown you that there are really significant changes that can happen just by changing the sperms. So there really must be something to find. And that leads me down the path of thinking that what we actually need to be doing is integrating measurements, combining measurements, but also designing new tools to get greater insight into those things that I've said a sperm needs to do. At the moment, we tend to look on a very high population-based level on some gross features of a sperm, when really we want detailed measurements of individual sperm, non-invasive, that allows us to gain these sorts of insights. I'm a mathematician by background, so I would say that one of the ways we could do this is combining mathematical modeling with imaging of sperm to provide part of the answer. So I'm going to spend a little bit of time in the next few slides just talking about what that means, because I'm sure a lot of you have heard of mathematical modeling and actually aren't quite sure of what that means in practice and how it can help address some of these questions. So to start off, I want to talk a bit about the flagellar waveform, so how sperm actually propel themselves and move. So I don't think it's controversial to say that motility is probably the biggest natural source of sperm selection. At the very first slide, I showed you that we start with 100 million sperm, and by the time we've got past cervical mucus, we're down to 1 million. That is a gigantic selection of sperm. In order for that to happen, sperm have to be able to swim. If they can't swim, they can't reach the egg. Natural fertilization just can't happen. It doesn't matter whether they're boundary following or chemotaxing or doing any other sort of navigation. If they're not moving, they're not getting anywhere. So starting with that idea, thinking about movement, thinking about motility, you then need to think, well, what's causing motility? So a lot of systems that talk about motility, whether that's manual analysis by WHO or it's CASA analysis by computers, really think about sperm head motion as being what motility is. But the head isn't moving. The head is being propelled by this tail that's responding to the environment. An analogy I like to use is if I'm in a river and you're watching my head and you're watching me move down this river, if you just watch my head, you don't know if I'm Michael Phelps swimming incredibly quickly and efficiently, or if I'm drowning and just being carried away by the current. You need to know what my arms are doing. You need to see the rest of my body in conjunction. It's the same with sperm. You need to be able to see all of that detail of the tail to be able to understand all of these things that we've been talking about. So whether the signaling and the ion channels are working in sperm, that's going to govern how that tail moves. Whether the metabolism is correctly working, that's going to govern whether sperm has the energy sources and is able to distribute that energy along the tail in order to move in the way that it moves. Those things are kind of fairly obvious, but there are other things as well that tail motion can give us insight to that might be less obvious. One is the fluid environment. I'm sure you've all seen pictures of sperm swimming in a high viscosity fluid versus a low viscosity fluid like IVF media. Sperm swim either with a lot more side to side yaw or far straighter. And that's purely physical because of the fluid environment. The way the tail moves also gives us insight into morphology, so ultrastructure, DNA compaction, because small changes to the sperm head have a much bigger effect on the tail by the hydrodynamic forces that are applied as the sperm moves. So just by looking at the tail, we are automatically getting a lot more richness of information versus just thinking about motility as how is the head moving. So in Birmingham, what we've spent the last few years now working on is basically ways in which we can track that tail motion and use that to gain insight into what the sperm is doing. So we created this software package called FAST. It's currently free to download and use. So it's a research tool currently but we are always looking for clinics who are interested in testing it. We are currently running a clinical trial in the UK, and then we have a number of international partners thinking about how we can use this software in the context of treatment to better predict outcomes. But what this software does, it takes negative phase videos of sperm, like this one. It could have 40 or 50 sperm in the image. I've just shown you one so you can see what's going on. And just like CASA, it finds the head, tracks the head, but also it extracts this waveform of the tail as it's swimming. So from that, you get to see the lovely variety in patterns that sperm make, whether they're in low viscosity fluid, high viscosity fluid, or whether they've been hyperactivated. You see the big change in flagellar curvature. But it also allows us to use mathematical modeling to understand things like what's the power being exerted by that tail, by that individual sperm as it swims. So what's the metabolic output of that sperm in a non-invasive way that we can then do on a population level to gain insight. We can use this on animal sperm. So this is a study by Gerhard van der Horst looking at ram sperm. So here they were all stimulated using procaine hydrochloride, and they get this lovely star spin pattern as they swim versus this very straight pattern that they swim with unstimulated. But the key is that tracking sperm is only part of the answer. So tracking sperm is great, it gives us data, but really we need to look at that data in the right way. It's not just a case of saying we have 100 sperm swimming quickly. We actually need to use this data in a way that actually gives us greater insight into those things that we've been saying about what a sperm needs to do. And really that's where mathematical modeling comes in. So I will show you one equation on the next slide, but don't worry, I'll explain it. Modeling is essentially the idea of using physical principles to write some equations. We can then put those equations into a computer simulation and then try and get some results out. So, for example, this is a study by a former PhD student of mine, and she really had the question, how does that little end piece, the tiny little flappy bit at the end of the tail, how does that impact swimming? Does it actually matter? What impact, what role does it play? That's something that's very difficult to do experimentally because all sperm have this little end piece. You can't really see it because it tends to be only a couple of nanometers wide because you're down to singlet microtubules at the end. So it's very difficult to work with on live swimming sperm, but using a mathematical approach, we can do simulations and we can see what's going on. So we began with this, which is a schematic that you're probably all quite familiar with. So here we have the axoneme, just like the previous speaker talked about. The axoneme runs all the way down the principal piece with this 9+2 arrangement of microtubules. And then in this final section, the end piece, you lose that axoneme structure and you end up with just these singlet microtubules that are just moving past each other. So really the question is, how can we understand what's going on in that final region? When we think about how the tail moves, it's moving because of those dynein arms, again, that the previous speaker showed, are pulling on these pairs of doublet microtubules to create bending. When you get down to the end piece where you've lost this structure, you don't have any active bending. So there's nothing for, there's no structure for dynein arms to be able to pull on. So this bit at the end is basically just flappy. It's not actively trying to bend in any way. So Cara, my PhD student, took that and basically turned that into this, which is a little mathematical model, and basically said, what if we've got a whole section of tail that wants to bend, and then we've got this little flappy bit at the end that we parameterized with this little L, and we say that bit is just not going to actively bend, it's just like a little piece of elastic at the end. So this is the one equation. What she turned that into was what we call a model, and that basically says that the curvature, so the amount that the tail bends, is related to the amount of bending force because we're in a fluid, with the amount of bending because the tail wants to bend. And we can solve that for a number of parameters. So we have this parameter S, which basically says, are we in a non-viscous IVF type situation, or are we in cervical mucus? And then we have a little parameter L that is the length of the end piece region. So physiologically, L would be about 5% of the total tail length. And what we can do now is we can just chuck this into a computer, we can solve this equation, and we can say what happens if L is 50% of the tail length? What happens if only 10% of the tail is actively bending and the rest is a big floppy region? How would that swim? And we can do a kind of systematic assessment of what's going on and kind of really test it. So 2 examples, one where we have 5%, that's physiological, or one where only half the tail can actively bend and one half of it is just big floppy elastic. So we have our question, how does the tail change swimming? And this is the answer. This is the sort of pictures we get out. So one, here we have no end piece. So all of the tail wants to bend and you get a waveform that looks like this. And here, this orange section in the end is an end piece and it doesn't bend. And you get this much more streamlined pattern, even though streamlining doesn't exist in the context of sperm, but this smooth pattern towards the end. And actually you notice that this sperm is swimming faster. So over these number of beats, it's swimming a whole, almost 2 heads further on. And actually we can do that for a whole number of different lengths. So this is our L. And what we see is that there's always an optimal velocity and an optimal efficiency of this end piece. And it just so happens that the physiological length of that end piece is pretty much where the optimal length is. And we've been able to test that and show that using mathematical modeling. Another interesting question when we're thinking about sperm is how do they actually generate their beats? So all sperm swim, well, lots of sperm swim. And when they swim, they're generating a beat. That beat is coming from some combinations of forces of these dining arms. But if we can understand a model of how they generate their beats, then we can use that model to probe how sperm are moving between different motility types and then trying to understand what impacts those motility types and how we can use that constructively. So the simplest model for how sperm control their beat is what's called curvature control. So basically this idea has been around for decades and decades, since the work of Brokaw, really. But the idea is that a section of the tail just wants to bend in one direction until it reaches some critical curvature, and then it wants to bend in the other direction. So each section of tail is just trying to bend like this little rod, and then you can build that up in lots of sections until you have a tail. So what we end up with is a model that looks like this, where we start with a flat tail, and you see each of those sections are trying to bend. And eventually what happens is it settles down into something that's nice and stable and starts to look like a sperm, just by applying this very simple idea along the tail. And if we use that concept for a whole load of these parameters that I've talked about, what you'll see is that that very simple model, that very simple idea, can give you sperm that look like sperm that are in a high viscosity fluid. So with that tight waveform with high curvature towards the end. They look like sperm that exist in a kind of low viscosity fluid, so with lots of side-to-side motion. And then you can get sperm that look like they're hyperactivated, so with really increased curvature in that first part of the tail, just by keeping that one very, very simple idea. And what that allows us to do is then categorize and quantify different phenotypes of sperm. So rather than thinking, oh, I've got 1,000 sperm that I've assessed in this sample, we can look at proportions of different sperm phenotypes. And then instead of thinking about sperm as being this big, complicated set of patterns, we think about it as just having 2 parameters. And that allows us to do a great data sweep and see what's going on. So just to wrap up then, giving you a whole load of things, but the things that I'd like you to take away from this talk is that I hope that you agree that we know that men affect outcomes. And for the benefit of all patients, but not just patients, when we look at all the data now linking sperm quality and male health more generally, we really need to improve male assessment. So the question is, where do we go from here? How do we translate a lot of these ideas into something that's more meaningful and more useful in a clinical environment? Well, first we need to really identify which sperm factors are meaningful for each treatment, and then which sperm factors are meaningful for selection. And really, the key problem is that most of the methods that already exist either lack outcome linked data entirely, or that data shows that they do not have good links to outcome, and therefore people don't talk about the data so much. And that's problematic. Really, all of these things only matter if we can show a link to outcome and use them in a constructive way. So we really believe that our mathematical approach, along with data, along with outcomes, has good potential to try and uncover some of these answers, but that's exactly what we're trying to do, and we're constantly looking. So happy to take any questions, and thank you for listening.
Thank you, sir. That was really the one of the most interesting thing I have seen relating mathematics to the infertility in the sperm. So thank you for your presentation. We are open for questions. If anyone have it, so you can post it in the chat or you can directly speak. Thank you.
I think, thank you, Murik. This is again a pleasure to meet you here, and your work. I have seen that since so many years you are doing work on that sperm movement, how it is swimming. So I think there are two questions sent by Dr. Hetal Sudha. Dr. Hetal, can you ask directly? You are here only.
Hello. Yes.
Yeah, please go ahead.
Yes. Yes, Dr. Murik, it was really very nice to hear from you about all of the things. I have two questions I will club together. The question is, one of the slide you said, and I really support that slide because I have seen the several publications saying advanced age, the female age is helped by PIXI more than the younger ones. Is there any explanation to this? I mean, how does it help advanced age female age? PIXI helps in better outcome. And the second question is, does this all mathematical modeling of the sperm movement and all, does it help in identifying the DNA integrity of the sperm? Thank you.
Right, two really good questions. So on the PIXI question, our best understanding of it is that really what's going on is that eggs. I mean, we know eggs have an ability to correct for some sperm issues. So good quality eggs, young eggs that are high quality, can mitigate a lot of the problems that you see with older sperm. They can correct some of the DNA. They have the ability to deal with all of these problems, largely because they don't have a lot of problems of their own. Whereas for the older women with the worst quality egg, the problems just become insurmountable. So they have a lot of their own issues that they have to sort out. They're then trying to deal with sperm factors as well, and basically it's too much for the eggs to happen. We suspect that this is actually some of the reasons why a lot of tests regarding sperm impact in IVF haven't shown any signal previously, because a lot of tests are done on donor eggs and sibling eggs with sperm, with the idea of you're then controlling for any egg factors, which makes sense. But the problem is the eggs are then so good that they're mitigating all the sperm factors, and you lose the signal. So actually, we're tending to think that more now when we're looking at sperm factors, we need to be thinking about using older, worse quality eggs, as that's where the signal is. In terms of the use of mathematical modeling to identify DNA integrity, it's a really interesting question. There's lots of bits of what we've done. I've not shown you it today, actually, but I've done a study that I'm currently writing up, looking at how slight changes in head size change waveform. And what we actually find is that, slightly surprisingly, slight changes in a head actually change the speed at which tails beat. So the idea is that then maybe that shows some sort of mechanism by which you could go the other way. We've done some studies looking at flagellar waveform parameters and DNA integrity, and we haven't measured by tunnel, I should say, and we haven't shown any great correlations. But I think this goes back to the idea that actually a lot of these measurements aren't particularly useful in and of themselves, and are actually measuring something slightly different, and it's useful having multiple measurements of the same thing. Because actually when in the Hub-Select trial where we did multiple DNA frag tests, we did tunnel, comet, halo, and aquamarine orange on the same samples, and we showed all of them are measuring things that are slightly different because none of them correlate with each other either. So there's a lot of kind of open questions there for me that I think remain to be answered.
Thank you.
I think there are so many diagnostic tests are available for diagnosing these kind of DNA fragmentation and ROS. So many tests are available. But actually, when we are doing live ICSI or something like that, we are going to do that IVF. Then how to select? That is the big challenge, because after doing that, these kind of diagnostic tests, we don't have any answer for that in live when you are performing ICSI. How to select this pump? This is the main challenge actually, but definitely there are some devices they can help us to reduce that kind of the time, because that sperm selection done by these devices, it will be, I think, will be better quality. At least you can select better sperm, and there will be less that abnormal sperm in that particular outcome what you will get after the preparation or like that. So,
definitely. And
yeah,
I think that also goes back to what I said at the start about yield. So with a lot of these preparations, so we've done some work on microfluidic preparation, and a lot of people, when they talk about microfluidic devices, their first question is, will I get enough sperm through? And actually that's not the question. The question is, will I get the right sperm through? If I only have 10 sperm, but those are the 10 sperm that would make it in natural fertilization, then you can take those 10 and inject them all into eggs.
yes, you're right. Absolutely right. Any other questions, Siddharth?
Sir, I have one question. Sir,
Yeah, please.
one of the question you only covered that. One of my question was, does the head size and the midpiece size affect the flagella movement? So that would be the most interesting thing to know using this. And second thing is, as you one of your slide showed that you PIXI and ICSI comparison, when the age of the female was increasing, the ICSI outcomes were decreasing. So whether in that study the age of the male was considered or not, because I think that would be the one of the more important factor which we need to see. Because
Yeah.
and the another interesting thing would be comparing the male age and the flagella movement of the sperms along with the head and midpiece. That would be the complete study and the more interesting one to get the proper outcomes using this particular thing. So if you have some insights on this particular thing.
Yeah, so head size, mid-piece characteristics, both of those affect flagellar waveform significantly. What's complicated is actually, so I showed you a few of the parameters, I glossed over some of them, is that we have very little detailed physical studies on things like the stiffness of sperm flagella, the stiffness of midpiece, the precise length of midpiece. We're getting, I mean, some of the electron microscopy stuff now is incredible, but it's on very, very small numbers of sperm. And what we really need is better physical characteristics of some of these properties to be able to then do the modeling studies. Because the modeling studies are very good for uncovering mechanism and probing and testing things. But actually, if you're starting from the wrong set of parameters, then they're less good for actually taking it to the next stage. In terms of the ICSI study, yeah, in the West paper, West et al., who did the mechanistic analysis, so that was in human reproduction from the Hub Select, we have exactly the same graph, but against male age. And it does exactly the same thing. It's not quite as significant as the female age effect, but it is the same thing. It's worth saying that on the female age graph, if you overlay the SART data for aneuploidy rates versus female age, it basically they're basically the same graph. So, yeah.
Thank you, sir. If any other questions are there, please you can post it in the chat box or you can ask. Yes, no more questions then. Thank you for your wonderful presentation. It was great having you.
Siddharth, if we have a question for Akash also. Akash, are you there? Akash? Akash?
Yes, sir.
Yeah, Akash is there.
Any questions?
Any question
Yeah,
for
if I
Akash?
can
Because he
answer.
is still waiting for that.
Sorry,
Yeah,
sir.
please.
Yeah,
Please go ahead.
I have one question for Dr. Akash. Dr. Akash, all this rheotaxis models and chemotaxis models, I know, I mean, they are important for natural conceptions. But any insights of their importance on ICSI cycles, because like we are bypassing all the steps for ICSI. So anything you would like to comment on?
Yeah. Thank you, Mr. Jesh, for the question. As of now, it is not proven as such how much of a role these things would have to play. But as I'd highlighted in one of the slides, this may be a potential gap which is present when it comes to fertilization in vivo versus in vitro, because they have found that the rheotaxis sperm has a better chromatin stability as such as well, and a better morphology. Morphology is understandable because it may give the sperm the hydrodynamic advantage in terms of orienting itself against the flow and having a progressive motility. But then, even having a better chromatin structure. This is the kind of thing which probably indicates that the natural selection is playing a role even in these taxis movements as such. I do not know of any particular study as such which has been conducted or validated yet, which gives a significant difference as such. So these are avenues which are probably open for further validation. And there are micro devices already in the process of being manufactured, where they are trying to get the micro grooves, trying to get the sperms move along the surfaces, trying to change the size, various channel size based on them, how the sperm moves better and the selection can be better. But these are right now, if I'm not wrong, not available commercially and still in the research stage. So maybe in this next coming up decade, I know there are so many all maybes and so's, but in the next upcoming decade, looks something exciting in terms of what microfluidics can deliver us, including in terms of chemotaxis and rheotaxis over there.
I think same question was asked by Dr. Devan also. Is there any device where these all taxes are available in that particular device?
Right now, not there, but I think they are under, you can say R&D as such. I think Vaitsar had his paper also over there where they had said that the main challenge comes in fabrication of these devices as of now.
Yes.
Technology exists to a good extent, but then fabricating these devices and getting them validated and getting them validated to be approved for human use. That is where it is being lack. But there are many people who are working on it. I think Israel and towards the Western Europe are the countries where they are actively working under it.
Yeah.
So if you have anything to add in this thing.
Yeah, I think I have seen that there is one person, I forget the name of that person on LinkedIn. They have created one device. The name is Riotech, something like that. Riotech or Riotex. So it include all these two chemotaxis and Riotaxis both. And thermotaxis, of course, we can do on temperature that surface or something like that. So all the parameters are they covered. It is on LinkedIn, and I think they are already marketing this product in their place. It is in Egypt. I have no idea, but it is from Egypt, I think. So they are doing that, and they are promoting all these parameters they included in that particular device, Riotex.
Hello.
Yeah.
Yeah, Shukla.
Yeah, Dr. Akash, may I give a very hypothetical question that can the chemoattractants be made of some use in conventional IVF? Because if it can help increasing more better, to replace the ICSI,
Yeah.
how it could
Yeah.
be?
Thank you, Dr. Shukla, for the question. I think all these devices which are being supposedly made in the future, and this would help in all the ART procedures, not just ICSI as such, I would say, because now we have micro like sorting devices which are available, which definitely give a sperm fraction from which conventional IVF is possible, and even IUI if the fraction is good enough. So I don't see any reason why this should not help in IUI as well, rather than just conventional IVF. But of course, ICSI would be the first easier target. It would all depend on the sperm harvesting, how much we are going to have from these devices, all queries. But given the streamlining and increasing the efficiency of these things, they should be the way forward. At least they would help in a better selection of the sperms, at least better sperm selection, if not separation. And
I think there is
achieving the scales which are present in nature, I think that would be a real challenge if we can get to there, because few tens to hundreds of sperms are surrounding the OCC complex in nature in vivo in the oviduct. So if we are able to reach that level, then that means we are definitely improving our selection in terms of the sperms numbers which are required to fertilize the oocyte successfully.
There is one company, I forget the name, that it is from France. They are also working on that chemotaxis kind of things. They have added something that chemotaxis, something like that device in this particular devices. It is under that research, because they contacted me also regarding that, but it was not happened actually at that time because lack of that financial that support. So now again they are started last year, and they got the good amount from the investors, and definitely in future in 1 or 2 years they will be with that particular device.
I think one of the interesting which I found about chemotaxis is usually it happens, many of the research goes from animal models to human models. But if
Yes,
I'm not
yes.
wrong, for progesterone, it was vice versa. As a
Ah,
chemoattractant, it went from human models to animal models.
right.
I hope I have answered the question, Dr. Shukla, to the
Yeah.
extent possible.
Siddharth, any other question, or we can move?
Not right now,
Yes,
sir.
sir.
No other
Thank you.
question.
Yes, sir, we
Chalo,
can.
chalo, move. Priyal, please go ahead.
Yes, sir. Good evening, everyone. So, on behalf of Enronet, this is my great pleasure to welcome you all to today's panel discussion on motility matters. Clinical relevance and standardization of motility assessment in the lab. So, the session brings together diverse perspectives from the experts in the field, and it is my privilege to introduce our first moderator, Dr. B. Ananta Lakshmi. She's a consultant and senior clinical embryologist at Janani Treki Fertility Center, the first woman from India certified as a reproductive embryologist by the American College of Embryology and nationally recognized academic leader. Our second moderator is Ms. Amita Subramanian, senior embryologist at Cloud9 Fertility, Chennai, with over 13 years of expertise and renowned for her research at international forums such as AISHRE and COGI. And let me introduce our panelist, Mr. Paresh Makwana, the leader of IHPR and senior embryologist at Wings Hospitals, Ahmedabad, and recognized for award advanced biopsy, blastocyst biopsy work, and international faculty roles. Our second panelist is Dr. Vikramjeet Singh, Zonal Head of Embryology at Cloud9 Hospital, a clinical leader specializing in the management of quality of the embryology lab. Our third panelist is Dr. G. Rahul Reddy, Professor and Director at EndroCare Embryology Institute, award-winning consultant in embryology and men's health. Our fourth panelist is Ms. Kanimoji, Lab Manager at Dasadaran Memorial Hospital, Coimbatore, ensuring high standards and innovation in the embryology operations. Our fifth panelist is Dr. Shivaranjani, Consultant Senior Embryologist at Motherhood Hospital, Bangalore, and she published a researcher and recent ACE Conference Award winner. Our sixth panelist is Ms. Dipti Ripale, IVF Lab Director at Sri Aurobindo Institute of Medical Science, Indore, leading clinical embryology research and innovation. Our seventh panelist is Dr. Ram Dayal, Associate Professor and Lab Director at Chitkarna University, Neeland Hospital, Punjab, and is noted for academic and laboratory leadership. Our eighth panelist is Ms. Shilpa Marwa Shima, Chief Embryologist at Grace Fertility, Gurgaon, ART, expert with advanced proficiency in ICSI-PICSI and embryoscope technology. So we are truly privileged to have such an accomplished group with us and look forward to the valuable insight each will share. Thank you, and now I'll hand over to our moderator to begin the discussion. Over to you, moderators.
Thank you so much for the kind introduction, Pune. To start our session, I would like to share my screen. Is it visible now?
Yes.
Yeah. So, sperm motility, we have discussed the two lectures are over, and now we are in the panel discussion. So, sperm motility is a long been recognized as a cornerstone of male infertility assessment. Despite of its importance, one of the biggest challenge we face as an embryologist and andrologist is standardization. So, in this panel, we are going to cover the clinical relevance and the standardization across the laboratories. So we have a beautiful set of panelists, and they are going to give their insights on the questions follows. So to start with the first discussion, we are going to explore both the science and the practice. So how we are going to achieve reliable and reproducible assessment of sperm assessment and the motility is what we are going to do in the upcoming discussion. So to begin with, fundamental but very basic, yet often debated topic, I can say, how to assess the sperm motility. So in this topic, we have a discussion recently on embryo chat also. So I would like to bring about a clear discussion on assessment of motility, which is going to be the basic one. In the lab, I would request the panelists to participate. Anyone can answer. Which is the preferred method of choice? We have glass light, macless, and Newbar chamber. What is your preferred method of choice, or what is your practice in your lab?
So in my lab, please continue, sir.
Yeah. Dr. Shivaranjani, you can.
Okay. In my lab, we prefer doing glass light method, the wet mount preparation. And then it is recommended to see 200 sperms to calculate the sperm motility. And at least 5 to 6 fields has to be checked. So this is a recommended thing what we do in our IVF lab, andrology lab.
Thank you so much. Anybody? Any other panelist differ from this opinion? Yes, sir.
We are using the macular chamber to assess the exact concentration and motility.
Okay.
So my preferred method is macular counting chamber.
Yes, sir. Just do you want to elaborate on any method specifically, sir? How will you count? How will you proceed with the motility assessment using MACLUS?
If count is more than 20 million, then we count only third rows, sixth rows, and 9 rows and divided by the total number of rows counted. So we will get the sperm concentration. If count and motility is less, then we count the 100 squares and divided by the 10 number of rows.
That is for
So
sperm concentration,
yes.
sir. About motility?
Correct. About motility, just we are counting those spermatozoa who are going very forward and direction, those sperm considered as a forward progressive type A.
No, sir. That is about grading, sir. Just we want to know about the counting in the MACLUS chamber. Motility assessment in MACLUS chamber.
Yeah. So in the motility,
how many?
Minimum 3 rows we are counting and adding the total motile sperm and divided by the total number of spermatozoa counted. So is equal to percent of motile in forward progressive. In such manner, we are counting to all of these factors.
We do in glass slide methods. We generally count in a Z pattern. Actually, we will take from the top, we'll go by this way, and then in the bottom. Likewise, we used to assess 200 sperms in different fields. We use a counting that scale also. We have a counting scale, and we used to do in a glass slide method.
Anybody for Nuba? No. Why we can we assess motility by Nuba chamber? I think it is no, because we are diluting the sample there. So all the sperms will become immotile. Hence, motility cannot be assessed using Nuba chamber. Is there any difference in opinion? Am I right?
Yes, you are right.
You are right.
Absolutely.
So next we'll move on to the preference. Why do you prefer this glass slide or MACLUS, and what is the procedure? So many of you have explained the procedure. Other panelist, Dr. Deepthi, anybody is using MACLUS for motility? You can give your insight on your preference and why do you prefer that?
Dr. Lakshmi, thank you. We use the glass slide method and we are not preferring the MACLUS, as it is mentioned in the WHO criteria also, and the volume which we are loading on the glass slide is also important. We won't be measuring, but loading with 10 microliters definitely gives a better motility selection. We generally use the cell counting chamber. By using that, we used to count which is progressive, non-progressive, rapid and slow together, and once we get the 100, and we always prefer to go with 2 replicates in both the replicates, and we'll take the average. If the average is more than 10, then we'll definitely go with another slide, another 2 replicates till we get almost same in the both the replicates.
Thank you so much. And any other variation or which can alter your motility assessment? Any other variables?
Size also, it should be 22 mm. It should not go with the 18 mm.
Yes, definitely. So if we don't have this size cover slip, what will you do? Can you adjust the volume or anything else can be done?
We generally have this cover slip size, and because the even distribution of the sperm, and if you use a lesser cover slip slide, like 18 mm, you will be having different layers, and the identification of the motility will be, there might be some errors. So we always go with 22 mm only.
Yeah, thank you. So if you are going to, as Dr. Deepthi was telling about the size, if you're going to alter the size, again, the distribution volume is going to be different. Suppose if you're not having that particular size, you have to adjust your volume accordingly. Any other things you want to add on, Shilpa? Shilpa Marva?
Size is also important, I think. So as mentioned, WHO, within 1 hour, the first thing you have to do is the motility.
Definitely. Shilpa, anything to add on?
I just want to add on, I just want to cover all whatever everybody is using, either a glass slide method or by Mackler's chambers. The things we need to keep in mind is, yes, definitely the volume, because more the volume, multiple layers will be formed, and that will gives an incorrect report. Definitely volume, cover size, and probably I think Mackler is easy to count with Macklers. So initially, if we start with Mackler chamber, I think for the initial beginners, it will be easy, and later on we can go with glass slide methods. This is my point of view.
So for motility also, you will be using your Macklers.
Yes.
I have one more point to add, that we need to see the count also. Like sometimes what happens if count is less than 1 or 0.1 million, in many reports I have seen occasional motility is being given. But counting in those like sperm count, it is possible you just take one droplet to count just 40, 50 at least spermatozoa. It's not necessary. If sperm is not there, then you have to complete count 200 only. If you have 50 spermatozoa in that cover slip, for very less count, we can use bigger cover slip also, but at least count them on the slide. Then even if you get 50 spermatozoa, then we can have like, if okay, like 20 spermatozoa is motile and 30 are immotile, then we have 40% of motility. So this can be also done like when we are doing motility for those sample where count is less.
I think glass slide is better if we have low count.
Yeah,
It's
glass slide only.
Definitely.
Cover slip can be increased, like we can use the bigger cover slip also, like when count is very less.
Yes, I think if we have very less count or very occasional sperm, I think it's better to do centrifugation. Do centrifugation, then only do semen analysis. So we'll be very sure if how many sperms we are getting if we have to go for it.
Yeah, that
That
can
is
be
good.
also done. Yeah, but
That is good
only
for assessing count, I think, but motility will it not be affected by centrifugation?
centrifugation, it will damage if.
So if it is very less, you cannot give accurate motility.
Yeah, but giving in percentage at least gives better idea rather than writing occasional.
Go with number of replicates, at least 2 to 3 replicates we can see, and we can count as many.
Yes.
So according to WHO, as Dr. Deepthi said, we have to count in 2 replicates, minimum 200 to 400. Suppose if you're not able to do that, you can adjust that, but that is not going to be very reliable. For time, just for a understanding purpose, we can do that. But when you want to decrease your error, always increase your number. So that is the thing. Amita, can you take the next one?
So the panelists, the next question that we would like you to answer is, what are you really assessing in sperm motility? How are you really going to grade the sperm motility? Can you give us some practical tips?
So we'll be assessing the sperm motility as 4 grades: grade A, grade B, grade C, grade D. So A will be rapidly progressive. The sperm is forwardly moving at least more than 25 micrometer per speed per second. So grade B will be slowly, sluggishly motile. So that will be moving at least 5 to 25 micrometer per second. So grade C will be immotile, I mean non-progressive, twitching or moving in the tight circles, less than 5 micrometer per second, and immotile will be not moving at all. So
Okay,
this
so
is how we do it. Grade.
your velocity speed, how do you manually assess your speed? You're saying 25 micrometer per second and all that. So how do you actually determine it manually? Not everybody has a CASA in their Andro labs. So, how do we really manually assess it?
So in wet mount slide, we usually see the motility like forward moving. So at least for a longer circle.
Yes, sir. Yes, sir.
In fast moving.
Yes, sir. We are having, sir.
So we'll be taking it as a rapid progressive, slowly moving in a forward way. Dart circle, that will be the slow, sluggish motile. So non-progressive will be tight circles or just twitching or moving the tail along. And non-immotile will be not moving at all.
Yeah. So how do you train your juniors when they are, trying to learn semen analysis? Because sperm motility is something that is very difficult to start initially. Concentration, you can do it quite easily with the Makeler, but when you're doing it in a wet slide, it is a bit tricky for people who are going to start doing it. So what would be your a suggestion for them to?
So for the new juniors, it is better to use counting chambers actually. So to see one sperm and then moving, and then click it as rapid one. So A, B, C, D, 1, 2, 3, 4 like that. So if second sperm should be checked, and then that sperm, how it is going, moving forward like slowly or in a forward motion, and then click B if at all it is a slow. So it should be like that. So as Anant Lakshmi ma'am told, it is like 200 to 400 we have to count and then take an average. So rapid progress, all
So do
percentages.
you warm your slides? Do you mount your slides on a heated stage? Does temperature really matter for sperm motility? Anybody else?
I think yes, we should always use warm slides before doing the semen analysis. And I just want to add one point. If any new beginner is doing the semen analysis, his or her report should always be cross-checked or rechecked by the senior one. This is how I think they learn doing semen analysis. And definitely motility is a critical parameter to analyze in a semen analysis. So they should be very fast while counting the motile sperm. They should not be slow, and definitely rechecking makes them better in their further semen analysis reporting.
So how many samples do they see before they are validated? Do you have a log count or something like that?
Probably for them alone to give a semen analysis report to a patient, at least 100 samples should be cross-checked by a senior embryologist.
Okay. And what is your leeway, plus or minus, if you're giving 40%? And if your junior is giving, say, 55% motility, will you accept it?
See, if it is above 50 million count, I think plus minus 5 million is okay. 5 to 10 million is okay. If the sample count is low, it's below 20 to 25%. I think we cannot take risk.
Yeah, you'll have to retrain them. So the WHO says that a rapidly progressive sperm is one that moves more than 5 head lengths per second, and sperm velocity is dependent on temperature. So we have to make sure that a standard temperature of 37 degree is maintained to achieve, so that if there is no variations in sperm motility. As sperm hyperactivate, as temperature approaches 40 degrees, the stability of the stage temperature is very important as per the WHO guidelines. So as Shivaranjani beautifully explained, this is how we grade sperm motility. And with respect to sperm motility WHO guidelines, what are the cutoffs for sperm motility?
The total motility should be more than 42%, and the total progressive should be more than 30%, both rapid and slow progressive.
So the difference between 2010 and 2021, they say 40% in 2010 and 42%. Does this really matter? It's 2% difference. Does this really matter in deciding our treatment protocols or whatever plan?
So we have Dr. Rahul with us. Sir, you can tell your clinical decision making on this, sir. Rahul, sir?
Oh.
Sir, Vikram.
Hello?
Yeah, any other?
I think Dr. Rahul Reddy is there.
Yes, sir.
You can take his opinion, because it is really good questions about this. In WHO 2010, it was 40, and now it is 42. Does it matter or not? I think he can tell us about that, how the treatment definitely
Is
that, yeah. Will treatment affect this kind of motility? Dr. Rahul, are you there? I can see
Sir,
you
I
are
think so,
there.
sir has some connectivity issues, sir. I think so, sorry.
Okay.
We can
Anybody
carry
else?
forward and come back to sir.
Yeah.
Yes, sir.
Yeah.
Sir, progressive motility is matter, but the total motility is not matter, sir, because total motility include the type C also.
So,
Any other?
so if you have a total motility of say 10 million, would you proceed with an IUI? I mean, total motile sperm count is say 10 million. Would you proceed with an IUI?
No, ma'am, just we are considering total progressive motility, not total motility. In that total, we are adding 3 parameters. So if low progressive motility percentage higher, then no allowed to do for IUI is not ideal.
So you prefer your total progressive motility more than your total motile sperm count, is it?
Only we are considering progressive motility means
Okay.
to go for IUI.
Okay. Ma'am, you can take it.
So, as we are discussing about 2010 manual and 2020, in 2010, they have given only 3 grades. It is classified as progressive, non-progressive, and immotile. Dr. Vikram, your insights on this? Can we move back to the same method of grading the motility as 3? Progressive, non-progressive, and immotile.
Sorry?
Can we move back to the grading of motility as 3 categories? Because in 2010, we have only 3 categories. So, in what way these 4 categories help a clinical decision making?
So basically, if we have 4 categories for the motility, in the cases of IUI and IVF, wherever the motility is very much important, then it is a very relevant, because in that we can see how many are hyperactivated motile, like motility is more in those kind of the spermatozoa, because once we categorize spermatozoa on the basis of progressive, non-progressive, and immotile, we don't know how many of them are slow moving spermatozoa, which we all see in many patients. Like there will be the progressive motility, but you won't feel like the sperm is not rapid enough to reach to the fallopian tube or fertilize in the IVF. So we are not very confident in those kind of the spermatozoa. So having 4 parameter is always better. So at least we can vouch like, oh, in this kind of the sample, there is a more chances in IUI or in IVF. Or if we don't see type A kind of motility, like if only slow progressive and non-progressive spermatozoa is there more in the spermatozoa, more in that sample, then we can tell to the patient also that this kind of the sample is not very ideal for the IUI and IVF. We might have chances of fertilization failure or sperm not reaching to the fertilization site.
Thank you so much for that. Yes, we have studies on that. So why it is important? Because as you said, the ability to swim towards more efficiently through the female reproductive tract and reach the egg is based on that. As we have to elaborate lectures on this, so it is very, very important. So as you said, to navigate and reach the egg and penetrate and fertilize the oocyte, we may land up with fertilization failure if we have low grade A motility. So when there is no or very few spermatozoa with grade A motility, there can be complete fertilization failure. So is there any panelist opting for lower motility and conventional IVF, or you want to do IUI or directly go for ICSI? Any panelist opinion on this, please? Dr. Deepthi? Sir has joined. Rahul sir has joined, I think. Deepthi, Dr. Deepthi, you can say.
Actually, if the motility is very poor, actually the 4 types of motility comes into the picture, and it depends on the percentage of the percentage of the motility is there. If normally in any sample we can see all the 4 types. The problem comes when there is a lesser percentage of the rapid one and the more percentage of either slow or non-progressive. Then that is the point we need to decide where to go. If the count is good and the motility, overall motility is still there, but the rapid and slow, the percentages are differencing, that is the point where the decision making helps in us. If it is very less, and we also need to see the count and morphology into picture. If the motility is poor and the morphology is also poor, then I definitely suggest them to go for ICSI. If the morphology is still good and sometimes we'll go ahead with the sperm preparation and total motile fraction we'll see, then we'll decide whether to go with IUI or IVF.
Now sir has joined. If Rahul sir can throw insights on this.
Sorry,
Yes.
my signal was not good.
Okay.
Yeah.
Sir, so based on your motility, will you decide the patient for an IUI or IVF for ICSI?
Yeah, definitely.
The percentage you will based on the which you will decide on.
Definitely. According to me, I take 20% progressive motility as benchmark for IUI. So if it is less than that, I put them on treatment. In spite of treatment, if motility is less than 20%, generally I suggest IVF. If we can correct the motility to 30% range, then we ask them to try naturally for natural pregnancy.
Okay.
Ananta, can I ask one question to Dr. Rahul?
Yes,
Yes.
sir.
Yes.
So, Dr. Rahul, as you mentioned that about the motility 20% or 30% criteria, does it matter when there is the age is factor? Does it matter?
No, definitely for female, first in age factor, what we look at is female factor. If female factor is female age factor on the higher side, we generally go ahead with whatever gynecologist with the obstetrician's decision. If there is no female factor, then only we suggest IUI. Otherwise, if there is female factor, then whatever gynecologist suggests, we agree with it. If they decide for IVF, we ask them to go ahead with IVF.
Ananta,
Yes.
Amita, can you ask same question what you showed in last slide? That about that
I'm
percentage.
coming there, sir. Yes.
Yeah, please.
So we were talking about the WHO 2010 and 2021 values, sir.
Yes,
so we
yes.
were there. They had mentioned that the total motility is 40%, like the cutoff range, and in the 2021 it's 42%. Does this 2% difference make change your clinical decision making? Does it affect?
No, I don't think so. I don't think so. It's only a statistical value based on whatever study that is.
Okay.
But in spite of normal parameters, if there is no conception, anyway we have to take a decision. So clinically, this 30 or 32% doesn't make any difference. What is important is take-home pregnancy rate. Sometimes we see even patients with 10% motility, 15% motility getting pregnancy. But we just have to go ahead with what WHO guidelines says. We say it as percentile value.
That's what, sir. So in your opinion, would you consider total motile sperm count or total progressive motile sperm count as your cutoff?
Progressive motile count.
Okay. Okay.
Thank you, sir.
So moving on to a case scenario for our panelists. The case is a male, 32-year-old, non-smoker software professional, and a female, Mrs. A, 30 years old, with regular cycles and no known female factors. They have been trying to conceive for about 2.5 years. And the semen analysis report shows a volume of 2.8 ml, sperm concentration of 18 million, progressive motility of 15%, total motility of 22%, morphology as per the Kruger-Strick criteria is 5%, and a vitality of 70%. So what would be your suggestion?
So as Amita asked, here I want to ask you whether the total motile sperm concentration is important or total progressive motile concentration is important. So in this case, when you calculate TMSE, it is around 11 million. It is when it is total progressive motility, it is 7.5 million. What will you make take that as important? Will you decide IUI for this patient or not? Sir? Vikram sir, Rahul sir.
I think that depends on patient also. Sometimes patient ask, but strongly we cannot recommend them to go IUI with this kind of the sample. because the count is like if we are having 7.5 million, then so yeah, there are a lot many spermatozoa, and we do require only one spermatozoa to fertilize the oocyte and give the pregnancy. But we have to check the probability also, like what is the morphology of the sample. If the morphology is good, then we can go ahead with the sample. If it is borderline, then we can suggest to the patient that IUI won't be good idea for you. So but at one chance we can give to the patient, in my opinion.
So based on female age, we can decide, I think, sir. Yes, sir. Sir, do you agree
Yes.
with that?
Yes, yes.
So here there is no female factor. Age of the female is 30 years.
So one more thing, like when we say there is no factor like involved for the infertility, that is the biggest thing in the infertility. If tubes are patent, if periods are regular, and if they are having regular intercourses like during then there is something like in the nature, like if we think sperm might be reaching to the fertilization site, but they are not able to fertilize. Then this is also one thing we have to consider. If couple is trying for the certain period of the time, and if everything is fine, and if they are not conceiving, then again it is an indication for infertility. Even though tubes are patent, periods are regular, sometimes in those kind of the patient we see, like even though if we have done IUI, failure is there, then we do the OPU, and we see the quality of oocyte is not very good. So, and one more thing is there, like sperm viability post insemination. So sometimes what happens, we see like good motile spermatozoa is there after post was 95% motility, but in some patient what happens, the very fast decrease in motility will be there in the media. Like if you keep incubating those spermatozoa, like after 2-3 hours only, you'll see the big chunk of pellet is being formed from the suspension also. So we need to, the 24-hour survival test is also a very good indicator for choosing the spermatozoa for IUI.
So total motile concentration, here you will consider that as important.
Yeah, it is,
For
but
an
morphology also we have to consider.
Any difference in opinion among the panelists? Dr. Sivaranjani Kanimoli.
Can I add something?
Yes, Dr. Shri.
I think we should try 1 to 2 cycles of IUI with such kind of samples, because there is no major factor female we can see. So I think if we can prepare, if we have total motile count more than 10 million or total progressive motile count more than 5 million, we can try 1 to 2 cycles. We never know if we are bypassing half the way through IUI. Maybe it clicks sometime, but yes, it is not very much recommended. We cannot get a very good, fantastic results, but at least before going for an IVF or starting a treatment, we can try with 1 to 2 cycles of IUI.
Yes. Can I add one more version?
Yes, sir. Yes, sir.
Yes. See, in this scenario, particular scenario where morphology is normal and count is just on the lower border, only progressive motility is on the lower side. So most probably this is a treatable cause. And according to our study, most common cause for low motility is infection, either epidermal infection or genital infection, seminal tract infection. So even before doing IUI, I suggest doing a semen culture test. I strongly believe that semen culture test, if it is positive, it's better to avoid IUI also. Because sperm, this bacteria can attach to sperm tail and it can travel.
So here we have not commented on round cells, as you said. So
yes,
we cannot.
No, in spite of whether there are round cells or pus cells, but at least before IUI, if motility alone is on the lower side, then it's better to do a culture test.
Okay, sir. That is a practical point. Thank you for
Yes.
that.
Yes.
So usually,
some post-culture test after putting the patient on antibiotic course, can we then go ahead with IUI or still not?
No, better. See now, if there is infection, positive infection in the culture, there is very high chance of high DNA fragmentation index. So then there is no point in doing IUI. And even if IUI succeeds, there is high risk of early pregnancy loss. So it's better to wait for negative culture, then go for IUI.
So one more question: if the culture is negative, would you suggest them to go for some antioxidant treatment for 3 months and try naturally, or would you ask them to go immediately for IUI?
No, generally we suggest them to go for treatment without treating them for 2 to 3 months. We never send them for IUI unless there is some other factor.
Because nowadays the lifestyle modification itself help them in conceiving naturally, because
Yes,
these are
yes.
younger couple and both of them seem to be on borderline, so there is no major issues. I think they can go away with some lifestyle modification or for 3 to 6 months, and they can see if they're conceiving naturally. In many of the patients we usually see these things.
Yes, doctor. Yes. So sometimes culture may be negative. See, culture regularly tells us whether there is any bacteria or not. Even if culture is negative, it can be fungal infection or it can be viral infection. So always, if culture is negative, it's always better to put them on anti-inflammatory drugs. What we do is clinically we examine. If epididymis looks bulky, generally we put them on anti-inflammatory medications along with antioxidants for 2 to 3 months. If motility is improving, then we ask them to try naturally. If in spite of that, if motility is not improving, then we suggest IUI.
Thank you for that, sir. Suppose if we are going for instead of IUI or trying repeated IUI, if you are going for conventional IVF in this case,
yes,
will you opt for conventional IVF or directly go for ICSI?
No, that depends on DFI value. We do DFI before IVF or ICSI.
Okay, sir.
If DFI value is normal, then both are equal for.
Any other person?
So do you do DFI for all your patients, sir, or
only before procedure, doctor? Any
before
whoever is planning.
IVF
Yes.
for ICSI?
For all IVF,
Yes.
you're doing DFIs.
You're doing DFI.
Yes.
So based on your DFI values, you will decide whether go for conventional IVF or ICSI.
Yes.
Thank you, sir. So any other panelist is supporting DFI before going for conventional IVF, or you don't want to proceed with the DFI and directly go for conventional IVF?
I think nowadays
Doctor?
DFI is must for all.
Okay.
Yes,
But
yes,
yeah,
DFI is must for all. And one more thing is there. I would like to add,
yes,
like
sir.
when we see the debris and like whenever we are preparing morphology slide, then we need to train our juniors also, like if they can see the bacteria because see, whenever we are staining the spermatozoa, bacteria also get stained with the similar stain. So it is not necessary that if a sample is having some bacteria or fungus, it will grow in the culture test. I have seen many times I am able to see the bacteria like streptococci or some kind of other bacillus, but the culture report comes negative. So that is also there. So we have to train our juniors also to see, and like whenever we are doing motility assessment in glass slide, that is a very best way, because when we are checking the spermatozoa at 40x, we at least can make it out like what is the morphology of motile spermatozoa, which that inside we don't get until unless we are doing ICSI. So while doing analysis, it is a very important part. Like whenever your junior is doing or you are doing, like we have many patients in which morphology is very less. So we prepare one wet slide preparation and we see at 400x to 600x that what is the morphology of motile spermatozoa also. So that is the advantage of doing motility in 40 in glass slide also.
Yes, sir. Definitely, I agree with you. And if anybody want to go directly for ICSI without conventional IVF and without DFI?
No, it
Any
is
other panelist,
generally
Dr.
not.
Dr. Ram?
Yes, madam.
Yes, sir.
I don't think anybody will answer it in other way.
because now it has become like a legal issue, doctor. Now one IVF failure, if they go to next IVF center, if they do DFI is on the higher side, then patient will start blaming the first center.
Okay. For practical reasons, yes.
Yes.
So you want to proceed with DFI and then decide on whether it is. So if you want, what is your cutoff for DFI and what is the test will you advise?
SCSA method.
Okay.
SCSA method, and my cutoff is 25.
Okay. So below 25, you will opt for conventional IVF. If it is more than 25, you want to go for ICSI.
No, basically, if it is less than 25, whether it is IVF or ICSI, both can be done. Only thing is, if it is on the higher side, then ICSI may not be suggestible directly.
Okay.
Then either we have to use microfluidics or testicular sperm if DFI is on the higher side. Either we have to treat the cause for high DFI and wait for the DFI to come down, or if it is a chronic infection and DFI is not coming down because of the infection, then we can use a testicular sperm. If it is DFI is slightly on the borderline, then I think we can use microfluidics.
Okay. But there is no clear-cut guidelines in order to proceed with testicular sperm for high DFI, I think.
No, there are papers supporting that.
Yes, sir.
There are papers
and against that.
Again that.
Yes. So based on your total motile sperm concentration or total progressive motile sperm concentration, choosing IUI or conventional IVF, we have discussed. So total progressive motile sperm concentration we are talking about. So as anybody want to comment on the cutoff? How much cutoff will you adopt for IUI and move on to conventional IVF? Below than this, I will move on to conventional IVF. Above than this, I will opt for conventional IVF, like that. Anybody want to say a cutoff? Yes,
If below the concentration 30 millions and motility below the 40%, then I will go for ICSI procedure. If above that, then I will perform IVF.
Below 40% is your progressive motility you are talking.
Yeah, total motility, not progressive.
Okay, total motile sperm concentration.
Yes, ma'am.
Okay. So less than 10 million, anybody want to go for conventional IVF or directly ICSI?
No,
less than
not
10 million.
less than 10.
Less than 10 million of
No,
your...
no IVF.
No. So everybody want to go for ICSI. Is there any difference in opinion?
Yes.
So here in this article...
Because it will be too risky.
It will be? Sorry, sir.
It will be too risky because, see, European and American work culture is completely different. Their cycle are supported by the government. So if any fertilization failure happens, then patient won't create any scene or any buzz. But in what happens in India, patient is funding their own IVF cycle. And if fertilization happens, then everything is fine. If it is not, then patient will create another issue that my sample was not normal according to the WHO and it was on lower side, then why you have done the IVF? Then you won't be able to justify your action also. So that is also one thing we need to take care of.
Yes, sir, definitely. So according to guidelines, it is not advisable. But in India, as doctor was saying, we should adopt according to our country situations and the patient need also.
So would you do half ICSI, half IVF if your total motile sperm count is good and you have
no half ICSI, half IVF are doing when count and all everything is fine. And if patient is having some idiopathic cause for infertility, so we are doing half and half. But if we see slight doubt about the motility, like if it is on slow progressive motility, even post wash also, then we don't do, because we have tried for few sites and there was no fertilization. And then I never tried after it, because it is unnecessary, just experimentation for the patient.
Okay, so an extension of this question. So does velocity parameters helps to us to make this decision? But not all of us have the access to CASA systems to assess the velocity. So what is your opinion on it?
So post wash, you can see what kind of the tail movement is there.
Mhm.
We also don't have CASA, but yeah, I have worked enough with the CASA, but it's not like you can't make it out. Yeah, it takes the experience. Like when you see the spermatozoa in wet slide, and you can say, no, this motility is good, and this is a hyperactivated or capacitated sperm's motility. But it is usually you can identify on the slide like after post wash.
Okay, so the literature says that kinematic parameters like straight line velocity and curvilinear velocity is very important to decide whether to choose between IVF and FC. If the VCL is greater than 65 micrometer per second and if the STR is greater than 40 micrometer per second, then IVF can be considered. So with respect to velocity, this is what the literature says. Anything lower than this, ICSI is the recommended method so that you maximize your success. So the next slide will be moving on to the genetic causes for poor sperm motility. What do you think are the genetic causes for poor sperm motility? Kanimani? Dr. Silvanchanu? Am I audible?
Yes.
You are
No,
audible. I think some network issues with. They're not able to unmute, I think. So anybody can answer.
Dr. Shilpa,
Yeah,
Dr. Deepti,
Shilpa.
what do you think are the genetic causes for poor sperm motility, ma'am?
It can be mutation in the genes response, of mitochondrial DNA or mitochondrial protein, or mutations. Can I answer?
Yes.
And mutations affecting the flagella protein and ion channels responsible for the calcium influx for the sperm motility. So, and then syndromes like maybe Cartagena syndrome, where the sperms are immotile because of the flagellar.
So when will you test for genetic causes? What is
If
your indication?
motility, usually in semen analysis, we'll check for motility. If at all it is less than 40%, we'll check for vitality. So if at all the vitality is more than 54%, so okay, when to check for the genetic causes? If the motility is maybe less than 20%, and the vitality
less
is
than 20%. Anybody else? Sir?
Yeah,
Dr.
any
Rahul, sir?
panelist?
Yeah. Generally, we do motilities for all patients with less than 5 millions. We ask for karyotyping and Y chromosome microdeletions. But if motility is on the lower side, completely zero, then we ask for CFTR gene mutation test. And CAT-SPER nowadays it's not easily available, but now some centers are doing CAT-SPER in Hyderabad also. And other genetic factors like primary ciliary dyskinesia and translocations, even those things. But if it is less than 5% motility and not we couldn't find any other cause, then we have to do genetic testing.
So the most commonly done tests are karyotyping and Y chromosome microdeletions, and also for CFTR genes.
CFTR.
So the commonly seen genetic conditions would be MMAF, that's multiple morphological anomalies of the flagellum. And primary ciliary dyskinesia and this dyskinesia of the fibrous sheath. So next one.
Yeah. So here we are having a case scenario based on motility. Already we have discussed this, but I want to add more questions on this. Just you're having a sperm concentration of 45 million per ml and volume is around 3 ml. Motility is 5% progressive, 10% slow progressive, 10% non-progressive, remaining is immotile. Full of round cells, as we are discussing about that in previous slides. So here, when you're having more of round cells, you have to subject the sample for any test you want to do additionally. That is leucoscreen. So here, it is positive. So any other panelist or sir, Dr. Rahul sir, you want to do semen culture for this case? Will
Yes.
this case improve with treatment or not? What kind of treatment will you advise for this patient?
See, factors which affect the sperm motility, not only infection, even it can be varicocele, it can be increased testicular temperature, or it can be severe hormonal imbalance. High estrogen, abnormal TE ratio also can affect sperm motility. So these are the things, and if it is infection with long-term low-dose antibiotics and anti-inflammatory medications and antioxidants, in 70% of patients, they respond with medical treatment and motility improves.
Yes, sir. Sir, what kind of drug you want to give, sir? Specifically treat the infection or general antioxidants?
No. In infections, common bacteria what we regularly see are Enterococcus faecalis, Klebsiella, and E. coli, and Pseudomonas.
Okay,
these are the common bacteria what we see. So if it is Enterococcus faecalis, generally we start with ampicillin 500 milligrams twice daily for 2 weeks. If it is E. coli or Klebsiella, then we put them on either ciprofloxacin or levofloxacin.
Yes. So specific to that particular bacteria. So general antioxidants, will it help in this case, sir?
General antioxidants will definitely help, but anti-inflammatory medications like Chimeral Fort or Chimeral BR should be given along with these medications.
Okay, thank you for that.
And if culture is negative, still there are pus cells, even doxycycline, simple doxycycline also will help us.
Yeah, thank you for that, sir. For antioxidants, do you have any specific combination or particular thing to
No,
say which is
nothing like that.
effective?
I prefer a combination, but I don't. I'll make sure that the antioxidant concentration is not on the higher side. Even higher dose of antioxidants will suppress sperm production.
Sir,
So, I generally prefer 100 milligrams of coenzyme Q10 and at least 8 milligrams of astaxanthin, along with the regular combination like selenium, lycopene, and zinc.
Yes, sir. Suppose if the patient takes it for too long, is there any harm? Or some patients without prescription, they will think that it might improve. So they will be taking for 6 months or 9 months. So will that make a change in the analysis report, sir?
No, lower doses will not change. If it is higher doses, definitely we have seen that with higher doses of antioxidants, the concentration and motility will go down.
Okay. So they were prolonging the higher dosage of antioxidants is also harmful.
Harmful.
Thank you, sir. Thank you for that. Any other panelist you want to do culture for this case? Yes, definitely we have to do. Is there any difference in opinion on this? So anybody is doing semen analysis without leucoscreen, directly proceeding with semen culture? Embryologist, Dr. Ram, sir, Dr. Vikram, sir.
Yeah, we are doing because sometimes it is not possible like to see WBC all the times.
So many labs we won't do leucoscreen also. So
no, if round cell is there, but I'm just saying like if for culture doing this leucoscreen is not important because it's sometimes we see the growth in the culture, but we won't be able to see the WBCs in the semen. So sometimes it will be there, sometimes it won't be there. And many times I have seen even WBC is present, but there is no infection in the semen, even though we won't be able to see the debris also. So if it is because of some immunogenic or some trauma also to the testis. That can be because of that also.
And we are doing semen culture and recommending semen culture in those samples where more than 1 million in particular sample. All sample we are not sending for the semen culture.
Yeah, some labs routinely do that. So any difference in opinion? Routine semen culture, do you advise for all the patients before IVF or ICSI? Dr.
It's better to advise for all patients to perform the semen culture to avoid any infection in the culture condition.
Okay, sir. So any other reason? Whether if your culture is positive, semen culture is positive, before IVF ICSI, you want to treat that and take up. Is there any particular reason apart from this? Dr. Deepthi, Dr. Shilpa?
I think yes, if semen culture is positive, better not to go for IVF if you are planning for that. It's better to go for ICSI. But yes, I think before starting or in the start, we should go with semen culture so that we can put patient on antibiotics as soon as possible before pickup.
So routinely you are doing it or not? In your lab, you are routinely advising semen culture
Yes.
before IVF?
Before IVF or ICSI.
Yeah, thank you for that.
So,
this,
quickly, if some of you could just mention the endogenous and exogenous source apart from leukocyte, what could be just put in it?
So there are many actually. Like first we need to understand why the motility goes down. So that can be attributed due to the mitochondrial DNA damage. Even spermochromatin DNA damage also decreases the motility. And third is in the production. So we need to find all those causes, like whatever is affecting any of those parts of the spermatozoa will have direct impact on the motility, as like from the like if we see the high temperature, like in the cases like where we see in case of varicocele, there is a high temperature. If a person is working high, like heated area, like if someone is working in hardware, some steel plant or something, or someone is using laptop most of the time, then also it will cause the increase in the temperature of the testicular. That will again increase the DFI and decrease the motility. Then
Finding
all
the
the
source, finding the
radiology
cause,
workers.
and finding the cause and correcting it if possible is what we have to do first.
Yes.
And all these lifestyle factors and radiation and environmental pollution, all of that history taking is very important so that we don't miss anything before we conclude things that their motility is poor. If something that could be done to correct their motility, we must make an effort to do that first before planning the treatment. Next slide.
Yeah, we are talking about standardization. So we were discussed about the method, procedure, everything. So now coming to the standardization, pre-analytical factors like abstinence, sample collection, how they are going to impact your motility. Dr. Kanimali? Dr. Deepthi?
Yeah, actually, we should go with the WHO guidelines. Abstinence should be generally we recommend for 3 to 5 days. And sample collection, again, there are a lot of factors which might affect the motility. So that is already in our history form where we'll be collecting all the information about the recent diseases and if there are any medications, and all the history has to be taken completely. Then only we can understand what might be the reasons for the poor motility.
Definitely. Any sample collection method or place or the temperature? Do you want to comment on anything?
Sample collection, again, they have to collect with the masturbation. If they are changing the method of collection by going with intercourse or using condoms, then it will definitely affect the motility. So we need to know the mode of collection. If it is masturbation, then we also advise them to collect the sample to give a complete sample. If they spilled or if they mix something in the sample, then here the history comes the very important part. We had to take proper history and we had to mention them clearly, the instructions how to color. And if any sample is spilled or if anything goes wrong, then they should inform us.
Definitely. So we have to make a note on this in our reporting itself. That is very, very important.
Other than that, one more thing, one very important thing is that, like mainly in the north, like wherever we see the winter season, that time room temperature goes down like 4 degree, 8 degrees, some places 16 degree. So that is also not very ideal for the sperm. So make sure like once the sample is collected, at least it has to be there at 25 degrees Celsius. If not possible, 30, 32 degrees Celsius, at least 25 degrees Celsius should be there. Because if sperm is exposed to the 6 degrees Celsius or 8 degrees Celsius of external temperature, like most of the lab, like even laminar flow, like if we are not keeping sample at 37, laminar flow, the metallic body, the temperature goes down till 14 degrees Celsius, even though if the room temperature is set between 22 to 25 degrees Celsius in the winter. So the temperature also we need to take care of, at least mainly in the winters.
Thank you for that, sir. Sir, I have a question on the standardization. So do you advise any kind of incubator is a must for andrology laboratory or a test tube warmer to maintain the sample after semen collection at 37? Suppose if you're not having, it is fine or not?
No, it
To
is fine.
standardize it. Yeah.
Yeah, it is fine, but it's better to have one small incubator because first of all, the room temperature is keep getting varied. So you don't know when you see the lesser motility, and since when we are doing motility in newer parameter, like when you are counting rapid progressive, then progressive, then non-progressive. In this kind of the motility check, the temperature plays an important role. Otherwise, the other form of the motility will go down, and you will have a what to say that incorrect report. So I think temperature should be uniform all across.
Yes, sir.
37 degrees Celsius, like some reports say it is detrimental. So you can set your incubator at 32 or 33 degrees Celsius. That is still better than 16 degrees Celsius.
Yes. Okay. So any SOPs you have to have your for even for simple semen analysis, what are all the things to be included in your SOP to standardize this analysis or motility assessment? Dr. Ram, Dr. Shivaranjani, Dr. Shilpa, anybody can answer.
Yes, there should be some SOPs for any semen analysis. There should be standardization, clear instruction to patients for proper sample collection. In my perspective, temperature should be maintained at 37 degrees Celsius, either in the incubator or at test tube warmer, and that should be analyzed within 60 minutes after collection.
Okay. Just about SOPs, what are all the things you would like to include in that?
That there should be proper collection of the sample. There should not be any spillage.
Okay. Any other thing?
So
time of examination you're mentioning about?
Within 60 minutes.
Within 60 minutes. Okay. So immediately after liquefaction, you will be having start examining the sample. So don't delay more than 60 minutes is your point, right?
Yes. And abstinence should not be more than 7 days. We always recommend, because then we'll be having reduced motility and probably more DNA fragmentation.
Yes. Thank you for that. So Amita?
So moving on to quality control. So do you do any quality assurance, or do you have any ways to implement QC with respect to sperm motility assessment in your labs? Do you do anything routinely with respect to just sperm motility?
We actually standardize the procedure. The grade should be calculated as per the WHO protocol. So next thing will be the internal assessment, internal quality control checks every month or every 2 months once. So
How do you do that?
Checking on the efficiency, how efficiently the andrologist or junior embryologist are performing the the semen analysis, mainly the sperm motility.
Is all your analysis always double checked by another person, or it's just one person who's doing it always?
So we'll be checking till 100 cases the person should do with the supervision. So after 100, if at all we're happy with the assessment, like how well they are doing, we can independently we can ask them to do. And then if at all if they have any doubts in the motility, they have any doubts like whether it is 50 or 40 or anything sluggish or not, then we'll go and cross check.
So yeah, how often do you cross check a validated andrologist or a junior andrologist?
So I will check more than at least 100 semen analysis.
No, after they are validated after 100 cases. After that, how often do you check their performance?
Usually often like weekly once or randomly we'll go and check. So if any essays are there, like it's randomly we'll go and check the motility and then we'll cross check whether it is correct or not and teach them if at all they make any mistakes and educate them.
Do you do external quality control with respect to sperm motility?
Yes.
How is it done?
So we have other person from the other centers. So and then we'll randomly will come on days and they will check. So it is better.
Is there any other statistically to check if you're meant for sperm motility? So you basically do a standard deviation for sequential measurements of difference in motility, and that is plotted in the graph with the mean value and 2 standard deviation limits are measured. And then you and then when you plot the values between 2 different technicians, then take an average and then you put it in a graph, and then this is a proper way of doing QC with respect to sperm motility. You can refer it in many journals, and
yes. So anybody from the panelist, are you doing practically this method, or you maintain any records of that?
Not sure how many of you actually do such a thing, or are you aware of such a thing?
No, generally, what do I follow that when we take the readings from 2 different technician or 2 different person, I see how much variation is there in between. Like if it is less than 10%, then it's fine, because sometimes what happens, sperm is a motile cell. So getting 100% accurate is not possible. Like, because if someone is saying like right now 3 sperms will be there in that field, and if some other person comes like after 20 seconds also, that there is a chances the sperm count will be sperm number of the spermatozoa will change. So but both the readings should fall under 10 to 15% of the error bracket, not more than that. If more deviation is there, then again third person has to check the sample, then we'll see accordingly, like if motility is proper or not.
Thank you for that, sir. So yeah, moving on to other.
So latest technologies, are there any new technologies or research directions that could further improve how we assess and utilize sperm motility? So AI is everywhere. So we have AI for predicting sperm motility also. And as discussed, microfluidic devices in Dr. Akash's presentation is also done. So what do you think is the limitations of conventional semen analysis in truly assessing your sperm motility? What are the limitations with our routine semen analysis?
Anybody can answer.
Subjectivity.
Yeah,
and interpersonal variations and the abstinence days. I suggest like 2 to 3 days of abstinence. So WHO recommends 2 to 7. So that also depends, affects the motility.
This is about conventional analysis versus CASA. So anybody want to answer?
So when we consider CASA, then if there are a lot of debris and all that can affect even pinhead spermatozoa also. It sometimes it may include in the study. So that is a biggest limitation between CASA and manual.
But in manual, the thing is that the variation source could be the even slide also, because whatever slide we use, sometimes there are greasing over the slides. So we also need to check that slide is also proper. And second thing is the temperature in variation in temperature. Then abstinence.
Yes, sir.
Then even duration also, like if sample collection time and assessment time it's been very long, and mainly in the case of high viscosity or when liquefaction time is more. So there we can expect little more variation in the motility, because we are waiting for the liquefaction to happen. Then if it is not, then we are using mechanical or enzymatically method. And same goes with the viscosity. So in those kind of the situation also, we might get inaccurate motility.
Yes, sir. Thank you for that. So we are talking about CASA. So can a lab balance cost and accessibility to the advanced motility assessment tools in a low resource setting? If yes, how can we manage that? Do you need CASA for all the labs? What is your opinion on that?
So CASA generally I don't recommend to every lab, but in a lab like only OPD is there, like primary IVF center where is no technician, then CASA can be done. The biggest thing comes that CASA doesn't, the morphology part is still cannot be done with the CASA. Only count motility is being done with the CASA. And if we have good motility, then that is a suggestive of vitality also. But the morphology, it is the major pain point in my opinion for the CASA. Otherwise, it would have been absolutely complete semen analysis machine.
Yes, sir. Thank you for that. So WHO compliant manual microscopy with proper training can be more effective. So it is not must. Basic phase contrast microscopy will also help you to standardize the protocol. So last session about sperm selection methods based on the motility. So what is the sperm selection method or the preparation which you advise for the low motility patients? Panelists, every one of you, you can give your opinion. We'll start with Dr. Shilpa.
It depends on the procedure we are proceeding for. If we have low motility, for IVF, I generally prefer density gradient. For basic ICSI, if the count is okay, if I can see good number of sperms, I can do direct swim up. I prefer direct swim up. If
motility, yeah.
If not possible, then definitely we'll go for a single density or simple wash, followed by swim up.
Yeah. Any other? Dr. Deepthi?
I'll go with density gradient.
Depend on the semen sperm concentration, sperm motility, and semen volume. We choose the density gradient accordingly.
So here it is low motility alone, sir. Rest of the things are normal.
Motility is if motility is less, then we can go for single density, only 80%.
Okay. Thank you, sir. Any other difference in opinion?
Yeah. If the debris is very less, then we can go for a simple wash technique. Because the count is already low. If we go for a density, then most of the sperms will be filtered in the density itself. So if the debris is very less, then we can go for a simple wash method also.
Thank you. Thank you for that.
So in case of conventional IVF, if your progressive motility is very poor, would you increase your concentration of volume of a process sperm to inseminate just because some progressive motility is low, or you would always inseminate a particular volume for IVF?
We'll go for ICSI.
Yeah, we'll go for ICSI.
You don't want to take the risk?
No, the thing is that there are few things to consider. If you are increasing your sperm count, then see sometimes what happens with bone number of the sperm, the chances of getting contaminants is also higher.
Agglutination method.
So, and second thing, So fertilization failure is also there. And third thing is, like once the count of spermatozoa increases, that will also increase the ROS generation. So even though if you get the fertilization, it is not necessary that you will get better embryo quality. So in my opinion, it's better to avoid it. You should not use more than 50,000 spermatozoa. Like once you are doing like 1 or 2 hours of incubation inside the sperm suspension, and if you are keeping it overnight, then 20 to 30,000, not more than that in any kind of the scenario.
So, as you said, the range is between 20 to 50,000 motile sperm, and the finite volume is recommended. And the volume could be from 20 microliter to 1 ml. So, as you all felt, why take the risk? Proceed to exceed the tradition. So, do you think new sperm selection techniques like microfluidics or zymote chips is going to help you in selecting better motile sperm in cases of low motility?
Yes, that depends on certain factors, like what is the count. If count is good, then we can try, but we have to see the volume. If volume is 2 ml, so we can use the half sample for the zymote. You can just see if you get any number of the
Have you found
spermatozoa.
any significant difference in your outcome in embryo with respect to blastulation rate or pregnancy outcomes when you have used zymote versus your regular?
Yeah. So in that, actually, we are not using for many patients zymote, but what differences I have seen, see, in zymote, the morphology improvement is not very high. In DG, we'll get the better morphology of spermatozoa, in density wise, I'm saying. But once you discuss about embryo development, the embryo development is better in zymote. If we are able to find the good spermatozoa, the same quality, same morphology of the spermatozoa, zymote versus DG, zymote will give better embryo quality.
So there is only difference with respect to embryo quality and not with pregnancy rates, is it?
Sorry?
Can I add? I think DFI is a good indicator for going either for zymotes or microfluidics with respect to normal ICSI. So I think patients with high DNA fragmentation, definitely these will help.
So most of us would do all these sperm selection methods only when there is high DFI, right? So the literature says that zymote is an add-on in IVF that can be used and seems to slightly improve the number of good quality blastocysts and pregnancy rate. But of course, further studies are needed. There is another school of thought that says that it does not directly improve sperm motility, but zymote focuses on selecting sperm with better morphology and less DNA fragmentation, which can indirectly impact fertilization and pregnancy success. So with this, we conclude. Thank you very much for all your active participation. Thank you, Dr. Lakshmi, for inviting me as your co-moderator.
Thank you, Amita. Thank you, all the panelists. Thank you, Ahira and Andronet, for giving us this opportunity. Panel discussion was wonderful. And over to you, sir.
Thank you so much to all. Yeah. Siddharth, Devan is there? Hello, Dr. Devan
No,
is
sir.
there?
No, sir. No, sir.
Devin sir is not there, sir.
No, sir. He's not there.
Okay, then I can thank you all of you. So thank you so much for joining. It was a wonderful, and I was thinking there will be very small panel discussion, but it took almost 1.5 hours to complete. I was thinking their own motility, how much question we can create, how many questions we can create, but it was fantastic. I think more than 15-20 questions were there. So it was wonderful discussion, and definitely we did a very good what you call that justification to motility, and we discussed too much thing about motility, tail movement and everything. Since this was our second part of motility, I think we included most of the things in this second webinar of sperm motility. And definitely, again, we are coming with so many other things. And next, Dr. Charudat is here, and I think he would like to say something about ISAR embryology. Recently, he closed that early bird what you call registration. Now you can take the permission from Dr. Charudat Joshi. He can allow some more time to register in the SR embryology.
Thank you, sir, for this opportunity. And on this platform, I would like to announce that whomsoever are our iHera regular members who are attending, they all are done under early bird registration only. Even if it is closed down, they should contact me. I can get it done. That is one thing. Okay, and this is just a privilege for iHera people. You have to show me your faces in iHera webinar, then only it is confirmed. Okay, and then I again welcome all of you to that Indore ISAR Embryology Conference. It's going to be a good event. Please make it sure that you should attend it, because these are the platform where you come to know about the each other and what is going on and what is happening. So I think this is a good opportunity for all of us. So once again, I would like to invite all of you.
I think there is one good thing is happening this time. There are 2-3 workshops are very good, and which are focused workshop. One is on ICSI also. You can get certificate also for the training for ICSI.
This is what is planned actually, because many junior embryologists when comes in contact and they always ask that can we get some ICSI training. So ICSI is not just injecting oocyte, aligning the pipette and handling, it's everything about the microscope. So we have organized that kind of workshop with a certificate also. After the workshop completion of that conference, you will be getting a certificate of ICSI competency. And similarly for vitrification also it is planned. And similarly for our OM pickup OPUET also, it is planned that they will have an hands-on experience on the simulator, and then they will be getting a certificate, which will be hopefully helpful. So these are some initiatives taken in this ISAR Embryology Conference.
So thank you all of you and all the participants and all my panelists, moderator, faculty, and of course our convener, co-convener, all of you. Thank you so much, and thank you UNS for supporting us for this webinar and RX events for organizing this event for us. Thank you so much.
One more line I just want to add here is, I think iHera should think in a bigger way to offer the services to all these national conferences to collect the questions of our audience and put it in the conference, because due to hesitation or one or some other reason, no one asked in question. But you see, now this discussion was going on. There are so many things which is unanswered and still need time to get the answer. There are so many queries. So probably we can give them this platform that, okay, you use this, you put your questions, we will put to the concerned expert and we will get it answered. Probably in this way we can help them out.
Yeah, definitely. Thank you so much.
Okay.
Then
Thank you.
Thank you. RX, a technical person can switch off the recording also. And
recording stopped.
Yeah, are we live?